we've been doing in my lab. Some experiments we've been doing in my lab, I guess trying to test these really exciting ideas around how complex systems might evolve through constructive neutral evolution. So I kind of got interested in this a few years ago now. I was working on a project that was being led out of C. Vanderson's lab in Sweden. They'd noticed they were sequencing Buchnera genomes, so Buchnera are bacteria that are obligate endosymbionts of aphids. And they found these conserved pseudogenes. So the arrows that you can see on my, you can see this point here, these arrows are pointing to positions of frame shift mutations. And the intriguing thing at the time was that they had a phylogeny of the Buchnera, and some of these mutations had appeared sort of 50 million years ago or more, and yet there didn't seem to be any additional mutations appearing. So they weren't really behaving like pseudogenes. They appeared to be under selection. And when we looked at what those mutations, or the location of those mutations and their context, they're all associated with either poly A or poly T tracts like this here. So this is sort of a highlight of this region here. And so you'd see sort of a single loss of a critical nucleotide here, creating a frame shift, but no other mutations. So I got kind of excited and said, hey, gee, this could be, I wonder if this is a form of editing. So we ended up looking into it. It turns out that it's not editing, it's actually RNA polymerase slippage. So what is that and why is that relevant? So polymerase slippage is basically where an RNA polymerase will come along and hit a region that's homopolymeric, so either a poly A or poly T tract. It loses track of the number of A's or T's it's reading, and you get either a you can get occasionally the correct number of A's added. So in this case, I've got drawn a 9A tract, but you can also regularly get frame shifts in the reading of the creation of the message. So they come out of either too few or too many A's. And this leads to a heterogeneous population of messages. So when we looked at the expression of some of these genes, so we did this by doing RT-PCR and then cloning of the subsequent the PCR products. We actually found that exactly that kind of heterogeneity. So this is a bit of a messy or tricky to follow table, so I'll walk you through it. So here what we've got are several genes that are frame shifted in Buchnera, and the genomic sequence has 10 A's, and to restore the frame one needs to add an additional A, so to getting an 11A tract. And what we can see in the population of messages is that say for this one here, mu E, we found a whole lot of them that had the frame shift in the message. So this is an uncorrected case, but then another a whole swag of them which had a restoration of the frame. So these ones could in principle be translated, and you can see there's quite a lot of noise here. So there's some that have fewer or greater numbers, but we can clearly see in all cases that some of the transcripts have slipped into frame and so should be translatable. We find cases as well where there is a homopolymer that's actually in frame. So this isn't a frame shift, but it's also slippage prone. So you find these products that take the message out of frame. So those would presumably generate non-functional products. We see some cases of kind of extreme slippage where sufficient numbers of additional nucleotides are added that you get a whole extra codon added. So there are a couple of cases there. And then when we took, we also took one of these Buchnera genes and expressed it in E. coli, because E. coli and Buchnera are close, relatively close in evolutionary terms, and E. coli is known to have slippage. So we're just curious to see whether a Buchnera transcript gene will slip in E. coli, and the answer is yes, it does. So we can see that here. This is the genomic copy, and we see at least one copy where we could see a slippage event correcting the frame. So we were able to also show that one of these were able to raise antibodies, and the protein that genomically looks like it's a pseudogene is actually expressed. So this is a figure just showing a developing aphid embryo, and there's a specialized region called the mycetome, which housed the Buchnera. And so this is this lights up when we do an antibody stain for an antibody we raised against the MURF protein in this case. So it looks like it's produced. So what this kind of looked like to me, and we sort of had some time discussing this, and at the time we wrote the paper, is that this looks very much to me to be consistent with a model that Cavallo and Gray published many years ago. So there's a paper from Michael Gray in 1993, which I presume most people are aware of in this group. So they argued sort of for 3 steps in the evolution of editing, and that was written, I think, around the insertion deletion editing that you find in trypanosomes. So they said first step would be that some editing activity would appear. In this particular case, we know that the RNA polymerase in both Buchnera and E. coli is slippage prone, and they have a common ancestor. So you could sort of say that equates a bit to the first step. Then the second stage is that some sort of mutation is enabled because of that background activity, and that can then become fixed through genetic drift. So I've just given a couple of examples of the types that I presented just before. So here's a poly-A tract. We've got a mutation changing an AAG to a synonymous AAA, both coding for lysine in this case. This one here doesn't particularly do much other than create a slippage-prone transcript. But if one has a frame shift instead, as we observed, then you actually, if this is a gene of some importance, you actually now require slippage to operate in order to recover the function of that gene. So once you've got that, then you get sort of into this third position where if that frame shift becomes fixed in the population, slippage would now be required going forward. So then you've got maintenance of what you might call a form of editing. In this case, it's slippage. So so if that's the case, it really sort of seems to fit this idea. Well, the only real difference between these two populations in terms of these tracts is their population size. So if you look at E. coli, the genome is largely free of these kinds of poly-AT tracts. There's probably selection against them because they're slightly deleterious, because only some transcripts will be functional. So we see no sort of, let's call it editing in inverted commas. In the case of the evolution of Buchnera, they go through repeated bottlenecks because the Buchnera are maternally transmitted from aphid mother to offspring. And so that continual bottlenecking kind of creates nice conditions for genetic drift to occur, and so favors the fixing of these slightly deleterious poly-AT tracts. So that's kind of what I thought was going on. We didn't really end up writing the paper too much around that, but it kind of ended up spawning an idea, which we've been working on in the lab, which is that if that's the case, we should be able to reproduce this in an evolution experiment. So what my student Alicia Lai did was set up an evolution experiment where we took an an E. coli, which we mutated, made it a mutator strain just so we could speed things up and see mutations happening. And then she did two things. So she made a bottleneck line where each generation she's selecting one colony from a special a predetermined place on a plate, just putting it into liquid culture and then pouring that onto a plate, spreading it out, and then using that to then collect the next single cell culture, and so on and so on. And then she did the same with a set of control lines where instead of just taking a small amount of material and spreading it so that you get single colonies, she just dumped a whole lot more. So the population size is going to be bigger, swirls that around, and we get large amounts of growth. So these guys are not really going through a bottleneck, and these ones should be going through a single cell bottleneck each time we do a transfer. Okay, so what did she find? So she did this for 50 transfers. And so what you're seeing here is genome data from 10-day intervals across a 50-day experiment. So we've got the number of SNPs here on one side, which corresponds to the bars, and the doubling time of the bacteria, the E. coli on the right-hand side. And so what we see with the SNPs in blue, these are the bottleneck lines. The SNPs are accumulating, so we're clearly seeing genetic drift happening, whereas in our control lines that shouldn't have bottlenecks, we're really not seeing an increase in the number of SNPs. In the case of the doubling time, that sort of stays pretty stable for the control line, but they're getting pretty unhappy over the course of the experiment. So the accumulation of mutations is reducing the fitness. So that sort of tells us the experiment is kind of working as you would expect in terms of the population differences between the two control and the experimental lines. So then we went looking in the genomes for accumulation of poly A and T tracks. So we sort of did a bit of a look before we, from the ancestor genome, see whether there were plenty of cases where if you had a single mutation, you would make a poly A or a poly T tract as the sort of length that we know the polymerase would slip on. And so what you can see is control everything sort of fairly stable. So this is actually double counted. So that shouldn't be, these bars shouldn't be quite this high, but the total genomic cases, and then the just the coding sequences. So they say fairly stable in the case of the control, but you can see very clearly accumulating in the bottleneck lines. So we've got something to go look at now. So what we then did was to look for evidence of slippage. We sort of started looking at a few genes and doing RT-PCR, then cloning and sequencing those. And so I'm going to tell you about one particular case we came upon that was good because we could assay it. So we found a frameshift mutation in a gene called RSC, which is codes for a regulator of the arabinose metabolism pathway. So what we already know from previous work that if you knock this gene out, then E. coli can't utilize arabinose as a carbon source. So we generated 3 lines. So we have a wild type, which just has a functional RSC. Then we took one of the mutations that we observed in our evolution experiment. So this is an RSC that's got a frame shift at a poly A tract. So just like we saw in the book Nera. And then we also just generated a complete knockout of RSC in a separate line. So we've got these 3 different lines. And so then we can see how they grow in arabinose. So the prediction is that the knockout shouldn't grow at all. And then this one should be quite happy. And the question is what happens with the frame shifted case. So here's sort of what you can see, this is sort of just an OD showing growth. So in orange is the knockout, and we see no growth of that line whatsoever. Here's the wild type growing quite happily. And you can see here is our frame shifted line. So it's got a delay in growth, but its doubling time is also significantly increased. So it's taking longer to grow. So that's telling us that the frame shift is clearly still got a functional, there's still a functional gene there in the sense that it's able to grow in these conditions. So we think that's indicating that slippage is being utilized for production there. So it's costly, but it's not lethal. Okay, so we played around a bit, and we wanted to understand what happens with slippage and protein production. So we engineered in some slippage sites along the lines of the things we've been observing into GFP, and because it's easier to assay the effect on protein. And so we've got three different cases. We've got a sort of an empty vector control here, and then three different cases where we've either got an insert that's not slippage prone, one that's in frame slippage prone, and one that's frame shifted, which should be non-functional when they're sitting in a loop here in the middle of, or sort of outside of the barrel that forms the GFP. And what you can see is here's effectively this is a control which has got a loop in addition to the loop, but it doesn't have any slippage, and that's sort of I guess what you would expect for a good functioning GFP in terms of fluorescence units. And you can see that's quite heavily dropped when you've got a slippage prone site, and with the frame shifted, it's dropped further again. So it's very clear that this is impacting protein production. We also kind of looked, just because GFP's got nice antibodies in those cases, we thought, well, what happens to the protein? And it does look like when we've got these, so, and this is the non-slippage case, and these are the two that have either a intact open reading frame or a frame shift poly-A tract. We are seeing a bit of diversity of protein product, so that's sort of interesting. Okay, so what does all of that mean? Well, I think that the system in the case of Buchnera has evolved very much along the lines of the Covello and Gray model for editing, even though this is slippage, so technically slightly different. And we can see that there's definitely a functional cost to this, as we could measure in the with the RSC assay. There's some evidence that we've got that, yes, you do get some alternative protein products, so those could be grist to the mill for new function over evolutionary timescales. So yeah, all of that looks pretty nice. But of course, we also wanted to have a look at editing, because that's slippage is one thing, but editing is slightly different. So we did a second experiment that we're still working through. We introduced a gene for called PPR56, which is a protein that's been shown to be an editing enzyme from Fiscometrella, a moss. So it's from the mitochondria of that moss. So it's a cytidine deaminase, which does CTU editing. So what we did is we introduced that into our line that is the RSC knockout line, it's a mutator line. And so this strain is unable to utilize arabinose, and we stuck the PPR56 onto a plasmid under control of this RSC promoter. So that means we can turn it on or off by changing the sugar source. So if we give it arabinose, it can't metabolize arabinose, but it would induce expression of PPR56. And because of the way the RSC promoter works, if you feed the bacteria glucose, that should strongly repress expression of that gene. Okay, so this is a separate experiment done by three people, James Love, Danny Maddock and Sam Aris in my lab. And so it's basically the same type of experiment as I showed you before, 50 transfers. And this time we sequenced the populations and the ancestor, and the whole experiment was done on Arabinose, so under conditions that should favor expression of this editing enzyme. And we're still partway through. We've sequenced the genomes and have done the assemblies. And so what we were curious to know was, do we see any mutations that are T to C SNPs? So they, if a T is changed to a C, then an editing event that's a C to U should create a reversion back to the ancestral state. So we looked for cases where there were T to C SNPs that were fixed in the population, where we could, by that we mean we saw the SNP in 95% or more of all reads. And then we would look at it further if we found that the SNP was present in an open reading frame, and if it was the SNP led to a non-synonymous change, so that would alter the protein. So in our control lines, we see no SNPs that fit that criteria, so none that seem to have gone to fixation. In experimental lines, there's 183, and a bit of variation depending on the line. So on average, about 18 of these cases. So then we've done two things. We just sort of did an initial screen, some sort of fishing, if you like, to see if there were any cases where if we do an RT-PCR, we can see evidence of an editing event. And then we're also currently looking at whole transcriptome data. Okay, so what do we see? As I told you, this is we've set it up as an inducible system. And so we can grow the lines in the presence or the absence of either glucose, which should repress, or arabinose, which should induce. So we did both of those conditions, and we grew them both of those conditions, and did a screen of RT-PCR and sequencing of the transcripts, either under expression or repression conditions. So the prediction is that if the line is grown in arabinose, and editing should be able to occur. So if there is a T to C SNP, that's where you'd expect to see a rescue event, whereas it ought not to be editing under repression. So we're hoping to see an example of that. So the reality is the data is a bit noisy, and I've cherry-picked the best example to show you. And it's this one here. So this is a case where we can clearly see editing. So I'll just walk you through what's happening. So at the very top here is the sequence of the ancestral genome. And so you can see it's got a T at this position here. And then we looked at line 4. In our line 4, we've got fixation of a mutation here from a T to a C. So that sort of fits what we would be looking for. Here's just a PCR and DNA sequencing of that, of just the genomic region, just to confirm that what we see in our genome assemblies is the same. So we see a cytosine at this position. And here's the induction. So we've got our PPR56 induced because we've added arabinose, and you can see there's a T at this position. So that's pretty clear evidence that there's an editing event. If we don't add any carbon source, so we sort of have an uninduced state, we still see that. But if we add glucose, it flips that back to C. So it looks very clear that this is dependent on whether what we see in the transcriptome is dependent on the whether the PPR56 enzyme is expressed or not. So I think I'm pretty much at the end now. So what we're currently doing is we're looking at the transcriptome data. So we've done whole transcriptomes both under induction and in repression, and we're looking to see, whether to what extent do we see editing having appeared, and we can compare our ancestor line to our evolved lines to see whether we're seeing the accumulation of or the appearance of additional sites that weren't present in the ancestor. And there's kind of several things we can do by looking at the transcriptome. We can look for error correction cases like the one I just showed you, where you're reverting back to the ancestral state through the editing event. But we can also look for what we're calling diversity, where there's some editing event that alters a sequence that into a new state that wasn't present in the ancestor. So that could both be a synonymous or a non-synonymous change. So I'd say watch this space. We're trying to currently prevent my postdoc from cooking her laptop by trying to do these assemblies on her laptop, but I'm in the wrong place to get it to the right computing. So hopefully that all works. All right, so I'll just end by thanking the people that did the work, particularly in my lab, and hopefully there's some time for questions. Thanks very much.
Thank you very much, Anthony. I think we have time for one or two very short questions. In the room, on Zoom.
I just had two short comments, perhaps. Yeah, so I really like that the slippage case. Can I be heard? Yes, I can be heard. I really like the slippage case because it provides a kind of, it's an unusual case because we see that the editing capacity here seems to arise from something that prima facie is a dysfunction of a polymerase, or at least a suboptimal activity of a polymerase under ordinary conditions. So that's kind of interesting that you can have constructive neutral effects emerge from this sort of neutral case. So from this mildly disadvantageous case, which is interesting. And I also wanted to comment that I thought the next experiment is super interesting, and I can't wait to see what you get out of it. So please let me know as soon as you have results from that second part. That seems like really promising and cool.
Right, thanks.
Perhaps we can take Kerry's question.
Hi. Yeah, cool stuff. I was wondering if you could phrase it in the terminology of CNE for me to help me with it. I guess I just kept wondering, so CNE has this buffer part that corrects for everything. So that would be your editing step. That's the buffer, right? And that happened after the slippage problem occurred?
Yeah, I guess right. So yeah, I think it's basically I'm trying to think how I need to word this, but it's effectively saying that there is something that happens that I guess alters the genetic landscape, or the potential for the landscape, right? So you've got some activity, which means that if you have a mutation, it doesn't have the effect that would be, the deleterious effect that it would have if it weren't for the presence of that enzyme. So the enzyme's effectively masking the deleterious effect by correcting it, if you like.
I guess what I got confused is that I had to go back and check what we wrote in a review. So it's an excess capacity or a buffer. And in your system, that happened later. Am I understanding it correctly?
No,
or did that happen first?
So the enzyme is present, but then the presence of the enzyme means that the genetic landscape that's non-lethal is different, right? So without an activity, certain mutations might be lethal, but because you've got the activity, the lethal aspect of those mutations is repressed, and the mutation is therefore tolerated, which means you end up with this possibility of a system that now requires that first activity.
So some slippage is probably inevitable too, right? I mean, you probably can't avoid slippage.
Yeah, I would say that's probably true.
If the buffer happened first, why do you need drift to fix those deleterious things?
It's just a game of chance, isn't it? you've got a neutral or near or perhaps even slightly deleterious thing that's not necessarily going to easily go to fixation in a large population. It doesn't mean it can't happen, but it's
yeah. So
less
it will
probable,
it will
right?
drift to fixation. I understand that, but also if you had some kind of excess capacity, like something that could fix these problems evolve first, then they could evolve in a large population because you've changed the landscape, like you said. You've taken things that will be deleterious and made them neutral. Sorry, I think I'm taking too long. I guess I was trying to understand it.
Yeah,
a
they
bit
definitely
better.
can evolve in a large population. You just would say that the cards are stacked a little bit against them going to fixation. You know, we know from studies of sort of what happens to individual mutations over time. It's just the chances of them going to fixation in a system that's got strong selection, where the population is large, is quite a lot.
Yeah, but the chances of them happening in
lot.
all small population are small too.
There's a chance of the mutation happening.
Yeah. Right.
Yes, that's right.
So it works
I mean,
out.
so we in our experiment we cheated, right? We used the mutator so we wouldn't have to wait for a very long time. But yeah.
I think that for the interest of time, we should thank again our speaker. Thank you, Anthony.
Thanks.
And move to the second talk by Tyler.
So Noah, would you mind keeping track of the chat for me, because I can't open it without,
no problem.
It being on everyone's screen. So let's make sure I have this up right. Can everyone hear me? Let's see if I can get this out of the way. Okay. I was asked to introduce constructive neutral evolution and then to talk a little bit about some of the work that I've done on it recently. So that's what I'm going to do, but I'm going to introduce it in a way that I've argued for recently that I think is helpful. Apologize to people who saw my talk yesterday. There will be significant overlap, but I'll introduce some new examples at the end. Okay, so this is just the structure of the talk. I'm going to first introduce constructive neutral evolution, and then I'm going to talk about some cases that I consider higher level cases of constructive neutral evolution. They're higher level in the sense that they involve structures above the genomic level. Okay, so these are our classic cases of constructive neutral evolution: gene duplication followed by subfunctionalization, protein binding, and RNA editing. I won't go through all of them. Many people will have seen some of the examples before. I'm just going to describe in possibly laborious detail gene duplication, because I'll use it as an example throughout the talk. So in a gene duplication event, the one depicted here, we have the gene. That's the black square with a level of activity L. It's a rectangle. It duplicates, producing overall 2L in level of functionality. And for the sake of this example, we'll assume that 2L is just as good, or 2L is just as good, if not no worse than L. Then over time, we have a slow accumulation of mutations in both the gene and its duplicate. At first, say, reducing the gene to 0.7L functionality, and then second, reducing its duplicate to 0.4L functionality, overall resulting in 1.1L functionality. In this case, we've seen both duplicates become essential for something that was essential for something that was happening with just one before. Okay, so this will be my running example, and I want to do something possibly annoying, which is start with a kind of analytic definition of what constructive neutral evolution is, and then move into an account of it. So to define constructive neutral evolution, we need some notion of comparative complexity. So in this talk, I'll say that X is more complex than Y, if and only if X has more parts or part types than Y. Then with a notion of comparative complexity, we can define the notion of a constructive change. That's just a change that increases complexity. So a change from a trait T to a trait T prime in a system S is constructive if and only if S with T prime is more complex than S with T. We also need the notion of a neutral change. And I'll say a change from T to T prime in a system S is near neutral or neutral if and only if S with T prime does not differ significantly in fitness compared to S with T. So sometimes what I define is, at this point, is what I call naive constructive neutral evolution. That's just evolution that's constructive and neutral. But I think to define proper constructive neutral evolution, we need a new concept or an additional concept, and that's the notion of essential complexity. So this starts by defining an essential dependency. That is, a trait A depends essentially on a trait B if and only if loss or reduction of function in B causes loss or reduction of function in A. You might prefer to call this a functional dependency. We can talk about why I don't like that term. Then with the notion of essential dependency, you can define the notion of essential complexity. And this is going to be a subset of your set of complexity in total, and it's complexity that's involved in an essential dependency. That is, if you unpack all of this, it's parts or part types that are affected by loss or reduction of function in other traits. When you have the notion of essential complexity, a constructive change, and a neutral change, you can define constructive neutral evolution as follows. That is, any evolutionary change that's both neutral and constructive of essential complexity. Okay, so let's just go back to our running example. Why is this a neutral change if I'm talking about gene duplication? It's neutral because we've assumed the whole way along that the gene plus its duplicate is still performing at least level of activity L, and level of activity L is all we needed. It's constructive in this sense because there's an increase in the number of parts and part types. And it's constructive of essential complexity because at the end of the narrative, at the end of the gene duplication event, if you were to remove either one of those genes, that would result in a negative consequence, because the other one alone would be unable to produce level of activity L. Okay, so it's constructive of essential complexity and it's a neutral change. All right, so we have a definition of constructive neutral evolution. But a definition really isn't all we want. We also want a sort of set of conditions under which we expect constructive neutral evolution to occur. So for the purpose of today's talk, I will describe what I take to be an analysis of constructive neutral evolution, or the set of conditions under which I expect it to occur. In this recent paper, I argued for a generalization of CNE into three parts as follows. So I say, I can just change the slide. I say any system S with entities A and B is evolving by constructive neutral evolution, provided the following three stages happen in the evolutionary narrative. At first, there's presuppression, in which an entity A, called the presuppressor, has the capacity to edit, suppress, neutralize, reverse, etc., subsequently occurring mutations in entity B. That is, at the first stage, entity A presuppresses a series of presuppresses mutations in B. In the second phase of the narrative, you have construction, in which there's an accumulation of mutations in B that are near neutral for S due to the presence of the presuppressor A. That is, in the construction phase, entity A actually goes about suppressing a series of mutations in B. At the final stage, which I call dependence, after sufficiently many B mutants emerge, the loss of the presuppressor A becomes deleterious, or in extreme cases, loss of A would be lethal, since the loss of A would lead to the failure to suppress accumulated mutations in B. So what I like about this is that it has three parts. It seems to me easy to apply, and the three parts correspond to three stages that we typically tell in evolutionary narratives about the events of constructive neutral evolution. So now I want to just argue that, in fact, this does cover at least the classic cases of constructive neutral evolution, and then to apply it to some further cases at the higher level. So let's just consider gene duplication and subfunctionalization again. I think subfunctionalization fits into this structure like this. The system we're considering is the genome. The entities or classes are a gene and its duplicate. In the presuppression phase, the gene is a presuppressor that neutralizes occurring mutations in the duplicate. Really, they neutralize mutations in each other, but we'll just tell this narrative in one direction. That is, in presuppression, gene A presuppresses duplicate mutations. In the construction phase, there's actually an accumulation of mutations in the duplicate that are neutral for the genome due to the presence of the gene. That is, gene A suppresses a series of B mutations. And finally, in the dependent stage, after sufficiently many reduction of function mutations in the duplicate emerge, the loss of the original gene becomes deleterious, since loss of the gene would lead to the failure to suppress accumulated B mutations. So really, this happens both ways, and in the dependent stage, really both are dependent on one another, but it's just easier to tell from one direction at a time. Okay, so in this case, the sort of order of operations is the duplication followed by an accumulation of mutations. And lastly, at the dependent stage, there's purifying selection against loss. Okay, so what I've done so far is introduce CNE sort of definitionally. I've tried to give an analysis of it that helps us describe it. And I've shown that, at least in that one exemplar case, it does fit the description that I've given. Now I want to apply this description to some higher level cases beyond genomics. Okay, so in this paper in 2018, Ford and I applied this to some higher level cases, specifically cases at the interactome level, at the level of transitions between levels, and at the community level. But I've talked about those things enough, and I want to talk about some additional cases. So recently, I tried to apply this at the organism level, so to organs, at the level of microbial ecologies, and at the level of culture. So obviously the culture stuff is going to be the most contentious, but I hope you think that it fits the mold in the end. Okay, so before I talk about higher level CNE, I wanted to bring up that, so Rose Novick in this recent paper, this element in the philosophy of biology and structure and function says the following: one of Stoltzfus's initial applications of CNE was to gene duplication, which has played an extensive role in the evolution of regulatory DNA and metazones, in particular of the Hox genes. This suggests the tantalizing hypothesis that CNE has played an important role in the evolution of metazone development. I think is probably true, but when I talk about CNE at the higher level, what I don't mean is something like upward causation, where we have CNE operating at the genome level, followed by some effects that propagate up to, say, metazoan development, or the development of organs, or the development of microbial ecologies, or something like that. What I mean is CNE, where the parts that we're considering are entities at that level, so entities at the level. That's not to say that this isn't a very interesting idea. It is. Okay, so the first thing I wanted to talk about was the liver. I think this is an interesting example because the liver has a lot of functions. It's also an interesting example because not all organisms seem to require a liver. So there's a question, at least there's a question for me, why organisms would have something that has so many functions concentrated into one organ that is not sort of, in an abstract sense, necessary for that organ. So I want to explain the evolutionary narrative something like this. There was this proto-liver, which I think there's good reason to believe was some kind of modified egg sac. And that proto-liver performed a number of functions that other bodily tissues performed. And because it performed some of these functions, it acted something like a presuppressor or an excess capacity in the system. This allowed other tissues or other bodily organs to lose the capacity to perform those functions. And over time, iterations of this process resulted in more bodily tissues losing some function, and the liver already being able to perform those functions, having those functions essentialized as liver functions. So that's the kind of narrative I like to tell about it, and this is how I think it slots into the analysis that I've given. And so I'll just read this. So the system we're considering is an organism. A, the presuppressor, is the proto-liver or liver. And B is all other bodily tissues, traits, components, processes, whatever. In the presuppression phase, the liver has the capacity to presuppress subsequently occurring mutations in all other bodily tissues. In the construction phase, the liver, in fact, suppresses a series of bodily tissue mutations. And in the dependence phase, after sufficiently many other bodily tissue mutants have emerged, the loss of the liver would become deleterious. So if you were the ancestor of all things that have a liver that had a yolk sac, and you were at the stage before your yolk sac was when it was just presuppressing the functions that were happening in other bodily tissues, then at that point, you could have lost your yolk sac to no effect. But once other bodily tissues have lost certain functions and the yolk sac must perform them, then you've essentialized the yolk sac, or you've made every other bodily tissue dependent upon the yolk sac, and that thing slowly evolved into the liver. Obviously, there's been secondary adaptation. I'm not trying to say the liver emerged ab initio from CNE, but I do think it's a plausible story for why some functions and processes have been localized to the liver. Okay, so I don't want to talk about it in full detail, but I also think that you can use constructive neutral evolution type narratives to explain the persistence of vestigial organs. This is the hip bones in a cetacean. It happens to be the case that the hip bones in the cetaceans are attached to certain muscles that are attached to the penis of those organisms in males. So people have tried to explain the persistence of that trait, the hip bones, which is otherwise probably a vestigial organ, by sexual selection for its role in penis function. I think there's constructive neutral evolution narratives that can be told about these kinds of things that just involve us assuming that at some point in the past, there was a contingent attachment of this muscle to the hip bone, which stabilized it in some way or other. But then despite the fact that there's been loss in every other part of the hip bone, this one piece can't be lost now, because to do so would have a negative effect on penis function. I think it applies to other vestigial traits as well. I just think this one's hilarious. Okay, so the next example I want to talk about is one of my favorite examples, and it's macrobial ecologies. So I think we can also have CNE at the level of macrobial ecologies. This particular image here is a achlorophyllous orchid. It's achlorophyllous in the sense that it doesn't produce any chlorophyll, or if it does produce, those chlorophyll are completely bleached. And it makes a symbiotic association with a fungus that lives in the soil. In fact, it can't live without that fungus. It gets some of it, or it gets all of the energy, all of its carbon compounds required for energy from its association with this fungus instead of from photosynthesis. So the question is, why did it end up like this? So selective narratives for this kind of situation often involve stating that once the orchid was getting some of its carbon compounds from fungal sources, it was able to jettison its expensive and costly photosynthesis genes, thus resulting in the adaptive spread of aphotosynthetic orchids. I don't think that's a very plausible narrative, and I think a constructive neutral evolution narrative explains it better. And that narrative works like this. The orchid was making some kind of association, perhaps facultative association, with the fungi in the soil, and it was gaining some carbon compounds from them. At that point, when some of your fungal carbon compounds are available through your association, when some of your carbon compounds are available through association with the fungus, any mutation or loss of photosynthetic capacity is neutralized by that association with the fungus. So this is how I explain that using the kind of structure that I've set up before. The system we're considering is a forest ecology. The presuppressor is the fungus. The entity in question is the orchid. And in this case, they're organisms. Obviously, that's what I take it to mean. That's why I find this case interesting, because they're at the higher level. So in the presuppression stage, you have the fungus acting as a presuppressor for mutations in the orchid's photosynthetic apparatus. In the construction phase, there's an accumulation of anti-photosynthesis mutations in the orchid that are near neutral within the ecology due to the presence of fungi providing lost nutrients. I think that's
Tyler, can I ask for a point of clarification here? Is that
Sure.
okay? Are we interrupting? Is that
Go ahead, interrupt.
Why? Thank you. So can you clarify the construction and the increase in complexity here? Because I guess to my mind, an increase in mutations isn't necessarily an increase in complexity. So I
can and I will.
Okay, wonderful. Thank you.
Okay, so what I've moved to talking about is, instead of net increases in complexity, just increases in the proportion of complexity that's in an essential dependency relationship. So the proportion of essential complexity. So suppose a photosynthetic orchid is facultatively associating with fungi in the soil, then in a sense, the structures responsible for that facultative association are non-essential, because at that point, the orchid could live without the fungi, and it would be fine because it has photosynthetic capacity. But once it's lost photosynthetic capacity, its association with the fungi in the soil has become essentialized. It's now an essential dependence. And there is work on facultatively associated orchids. So there's work that shows that there are orchids that facultatively associate with fungal carbon sources. And the ones that are able to make this facultative association and get some of their carbon sources that way do have relaxed selection on their photosynthetic apparatus. So what I'm talking about is an increase in the proportion of complexity that's essential, even though the overall complexity is going down, I'd say, because you're losing your photosynthetic apparatus or you're pseudogenizing those genes or something. The proportion involved in the association with the fungus has become essentialized.
Okay, yeah, I guess I'll save this for discussion, but I'm worried that this could expand our definition of CNE at lower levels to things that we wouldn't necessarily think of as CNE initially. But I like the move so far.
Yeah, we should talk about that later. Okay, so I think I've described this case in detail, so I'll move on to another one. So that was organs and macrobial ecologies. I just wanted to point out that there's another wonderful case of this in Monotropa uniflora. So it's not just orchids, there are other achlorophyllus flowers that this has happened to, and I think in a very similar way. So let's talk about culture, which is super contentious, and hopefully what I want you to get out of this is not there is cultural evolution and it operates by constructive neutral evolution, but rather there is a certain plausible analogy between the way certain cultural complexity emerges and the kinds of complexity emergence that happens in constructive neutral evolution. So I love this quote from Gray et al. They say, although complexity in biology is generally regarded as evidence of fine-tuning or sophistication, large biological conglomerates might be better interpreted as the consequences of runaway bureaucracy, as biological parallels of nonsensically complex Rube Goldberg machines that are over-engineered to perform a single task. Very punchy statement, and all I want to do is sort of extract the biological details from this and just reapply it to culture. So I want to say, although complexity in culture is generally regarded as evidence of intentional engineering, sophistication, many cultural phenomena might also be better interpreted as the consequence of runaway bureaucracy, as nonsensically complicated Rube Goldberg machines that are over-engineered to perform a single task. And I want to provide some examples of this. My favorite example is the Spelling Ace or spell check software. I think essentially that spell check software works a lot like an editing system in that once we have the ability to perform spell check operations within our culture, we tend to see the spread of edit requiring sites. And these sites are just our inability to spell certain words correctly at certain times. How does that work? Well, there's actually a lot of people find this to be quite interesting to study. So there's lots of people who've studied whether or not spell check has an effect on our ability to spell things. And the overwhelming answer seems to be, yes, it does. But the experiments work something like this. You take a class and you split them into two parts, one of which you give spell check and the other of which you don't give spell check to. So the one that has spell check obviously learns to spell with spell check and learns to rely upon it. And when you remove spell check, they become worse spellers. So if you say, let them learn during their class while writing their essays on a computer with spell check, and then you make them write their essays at the end of the class by hand, then you see more errors than if you did not teach them to use spell check in the first place. So this is a case in which I think some cultural level phenomena, namely our ability to spell or our reliance on spell check technology, is explained by constructive neutral evolution. Here's a weirder example. I think automated milking technologies and the way that farming depends on them can also in part be explained by analogous processes to constructive neutral evolution. So there's these things called automated milkers. This is one in which cows walk in to get some grain, and then they're stuck in these little pods where they get automatically milked and then sent back out later. So obviously this is a feat of engineering, and I do not think that it emerged initially neutrally. But once we have a farm that's in place that is not using an automated milker, and we introduce automated milking technology, then I think this can explain how individual farms and farming systems as a whole can become dependent upon these technologies, even though they aren't now necessarily resulting in any benefit. So my narrative for this works like this. You have a farm. It has the presuppressor is the automated milking machine, and B is, I say, a farmhand, but really any other parts of the farm. So in the presuppression stage, the automated milker has the capacity to suppress, in this case, neutralize, subsequently occurring changes in the farmhand or the whole collection of farmhands. In the construction phase, there's an accumulation of changes in the farmhand. Perhaps their working hours are diminished, or they're trained to perform different tasks, etc. And I think these would be neutral for the farm because we have an automated milker. People don't need to be spending their time milking. In the dependence phase, though, once there's sufficiently many other changes in the farm, so all of the farmhands are performing different tasks, loss of the automated milker would become deleterious. Even if initially introducing it wasn't all that beneficial, now removing it would be catastrophic. Okay, so I hope that you find this example at least mildly plausible. The last example I want to talk about is something perhaps more dear to our hearts than automated milking, and it's the duplication of roles in institutions. And I just want to give a quick argument that duplication of roles can work something like duplication of genes. So it works like this for me. So I want to explain the emergence of some complexity in bureaucracies the following way. In the pre-suppression stage, you have a task X that must be performed that's difficult for any given individual, I1. So someone, usually a manager, partitions the responsibility for completing X to two individuals, I1 and I2. Since the task is divided between I1 and I2, neither individual is ever tasked with the entire completion of X, so both individuals pre-suppress the other's loss of ability or authority to complete X. In the construction phase, as time passes, either or both of individuals would progressively lose the ability to complete X. Perhaps they forget how to do it, or they're deprived of the authority to do it. And finally, at the dependent stage, eventually, both of them completely lose the ability or authority to complete X, so can complete X only when, if cooperating. In this situation, I1 and or I2 have been subfunctionalized. And I think this is directly analogous to subfunctionalization of genes. Okay, so this is what I want the takeaway to be. If we adopt this more general analysis of CNE, then we can more easily see how it might operate at higher levels of biological type.
Right, we have time for questions. The chat is mainly about universities being interested in bureaucracies.
Universities are an obvious example.
So should I stop sharing so we can go back to this?
Oh yeah, that makes good sense. Yeah, Stephen has a question here.
Yeah, for the example from the cultural context, I mean, you have to somehow, starting from your earlier definitions, you somehow have to say how you translate certain terms from the biological context into the cultural context, like the concept of fitness and the concept of near neutrality, like in the example of the automated milking machine. I mean, the fact that then workers can reduce their working hours, in what sense is that not beneficial? I mean, how do you spell that out? Do you not need to make your kind of analogy a bit more concrete to argue that this is really?
I mean, I would need to make my analogy a lot more complete if I were trying to argue for something like there is cultural evolution and in many cases it operates by CNE. What I instead am trying to argue is insofar as we already believe we have notions of cultural evolution that we apply, then we should also believe that we can apply constructive neutral evolution in those cases. So essentially, I'm leaving it up to others to fill in what they think of as, say, a cultural notion of fitness, which I think is highly contentious. But don't you think there are some cases in which, say, institutions come to have more parts and part types in a way such that no one in the institution really benefits from this? The institution as a whole doesn't benefit. And at the end of the process, if some of those parts, some of those roles were removed, the institution would suffer.
I mean, it sounds very plausible to me. I think just in the detail, the devil is in the detail.
Yeah, it does depend on how you define, say, the fitness of an institution and what the system you're considering is. So is it the whole system of farming in the automated milking case? Is it all classrooms in the case of spell check, or is it just individual spellers? I'm not sure. I think there's many levels of analysis you might use there, and I'm not committed to any one in particular, I don't think. Sorry, that's a very squirrely way to answer that question, but cultural evolution is a squirrely topic. So.
One final question by Anthony.
Yeah, I like that analogy with culture. Obviously, we can all appreciate some of those bureaucratic structures. I guess one thing that's maybe slightly different in the case of humans is we might get stuck in a particular process if there is a process there, right? But we're still able to switch and learn something different. So it's not like learning or ability to change processes actually get switched off. So there might be an inertia in a sense, but it's maybe a little different from trying to unpick something in a biological system when that gets fixed. I wonder if you thought about that.
Yeah, I mean, the question here is just the power of intention. So even if we're just thinking about ordinary cultural evolution without thinking about constructive neutral evolution, then there's always a question of what the power of intention to bring about that same outcome is. So if you try to give a selective narrative for why some outcome obtained culturally, and someone responds to you by saying, well, actually, I think the people involved just brought that outcome to bear intentionally, then yeah. I would say in some cases, when the fitness cost, however you define fitness, of removing something is so significant, then there's very little that we could intentionally do to overcome it. So say you're a farming community and you've already become dependent upon automated milking to reach your milking quotas, then regardless of how you might like to give your workers back their jobs or to switch to a different system, if the cost of reducing, say, how fast you can output milk that day is too high, then you won't be able to tend your way into that situation. But yeah, I'm sort of aware that there is a big background problem about human reason involved in the cultural cases, as there should be.
Okay, let's thank again, Tyler. And the next speaker is Doolittle, and
I thought it was Alessandro.
yeah, I put the sport first, yeah,
Okay.
and we'll hear about cellular bureaucracy.
Sorry, am I next?
You are, if you're ready.
I'm ready. Can I share my screen? Let's see here. Can you see that?
Yep.
Okay, and can you see that?
We see the
Yes, but you're on
presenters.
presenter view. You've just
Can you swap displays on the top left.
Oh, I don't know quite how I can do. Let's see here.
maybe swap. Yeah, try swap displays.
At the top left, there's yeah,
Oh, swap display. Okay. How about that?
Great.
yeah, that worked.
Okay, good. Okay, so I'm getting confused by the switching of the screens here, but okay, I'll try it. I guess I really want to say three things here. One is to talk about the molecular level of CNE again, and secondly to analogize everything to putting the cart before the horse, as it were, which I'm drawing there. And thirdly, to try to rationalize a paper that Tyler and I and Joe Bolowski, a population geneticist, wrote last year at Dow. So I think that I generally agree with Tyler about how we define CNE. And I think both of us live in fear that Harlan will actually show up and say, that's not what CNE is at all, but maybe he won't. So as a grad student, which was about 60 years ago now, I worked quite a bit on suppression. So I'm just going to read what I defined as suppression. Genetic suppression occurs when the phenotypic defects caused by a mutation in a particular gene is rescued by a second mutation in the same or a second gene. The fitness of the organism is restored through positive selection. So there's a maladaptive stage involved in the notion of suppression. Genetics. So I think I worked a lot on that as a graduate student, and so I think I was sort of primed when Pat Cavello pointed out to me that they had a problem with explaining tRNA editing in wheat, that it maybe was a case of pre-suppression. So I'm going to define pre-suppression. It occurs when the potential phenotypic defects caused by a mutation in a particular gene is rendered neutral by an activity of the same gene or a second which was already present. The fitness of the organism was never reduced, but now the pre-suppressive activity is additionally maintained by purifying selection. And I think that's what I would call constructive neutral evolution. And the difference between pre-suppression and suppression then is that the former, suppression, there's a period during which the second mutation is being selected for, during which the organism is less fit than the preceding wild type. And for the latter, there is no such period. The pre-suppressive activity pre-existed because its component was selected for some other reason. It's an exaptation, or for no reason at all, in which case it's a pre-aptation, which I'll define in a minute. So through pre-suppressors, though pre-suppressors are now retained by purifying selection, they were not created by positive selection, at least not for their current suppressive activity. Suppression and pre-suppression generate the same complexity dependence, but the latter is more likely to happen. You might even claim it was inevitable, a genetic ratchet. So in terms of rationalizing it with the paper that Tyler and Joe Belowski and I just published last year, we recognize that any trait that is currently fitness contributing, so we're talking about the fitness of organisms in particular, although this does apply to different levels at which natural selection might be seen to operate. All traits can be either an adaptation, an exaptation, in which case they were fixed for some previous reason, or a pre-adaptation, in which case they were not fixed for any particular reason. And obviously, we're relying on the language that was created by Stephen Jay Gould and Elizabeth Verba a very long time ago, exaptation. And so I think that I think that their paper, Gould and Verba's paper, was pretty much about contributions to current fitness at the organismal level. So they really, even though they were dyed-in-the-wool hierarchists, I think they talked about just one level of fitness, level of fitness of organisms in this paper. And so I'm calling adaptation what they called adaptation. And I would include in that suppression in the normal sense that I just defined it. But there would be an increase in functional dependence. Then the suppressor is also necessary for the action of the original component of the cell too. So there's an increased functional dependence. And otherwise, I would analyze things as pre-suppression. So what we've done here is broken down what Gould and Verba called exaptations into two categories. Exaptation legitimately, which is to our minds a character previously shaped by natural selection for a particular function, and adaptation, which is co-opted for a new use, exaptation. And then we called the second category of what Gould and Verba called exaptation a pre-adaptation, a character whose origin cannot be ascribed to the direct action of natural selection. A non-adaptation is co-opted for its current use. I realized in preparing this talk that actually pre-adaptation is what we imagine actually happens with random mutations, in the sense that a mutation might occur, and then whether or not it's selected for depends upon whether or not it's actually co-opted for some kind of use. So pre-adaptations does include simple mutation, I guess. So maybe in order to separate them from simple mutations, I would say that it has to be a complex random mutation, but I don't really know about that. So I would say that likely many proteins are exaptations, really, because it's hard to see a protein arising just by chance. But RNAs are more often pre-adaptations, I think, particularly when there is accidental transcription, or even as in the example that Anthony Poole mentioned previously, some faulty transcription. So with either or with simple suppression, there's an increased functional dependence. That is to say, and I think I'm defining complexity in the same way that Tyler does, the number of components required to perform a function, that increases. But suppression is more likely because there's no disadvantage, no disadvantaged intermediate, no cart before the horse, as it were. So, and I was reminded of this paper that was influential for me as a undergraduate, no, as a graduate student, sorry, by Francois Jacob about evolution and tinkering, because I think that was about proteins, and it was about proteins being repurposed, and I think that's probably what happens with most proteins that are involved in CNE, is that the excess capacity that they have is perhaps a side product of some capacity that they were actually selected for, possibly an unavoidable side product. But I think that differently, I think that RNA is mostly about pre-adaptation. And so recently, very recently, a book by John Maddock, who's sort of my arch enemy, I guess, probably Anthony Poole's almost countryman, almost, it involves, I think, an incoherent definition of the word function, but that's a separate matter. Perhaps we can go into that if we want to. So one example of protein dependent CNE would be, for example, chaperonins. So we might imagine, for instance, that a chaperone, a thing that refolds a protein or fixes up a protein, basically, arose by suppression, let's say, for the first folded protein that made the cell most vulnerable to detriment. That it might have been a suppressor for that, but now then, because it exists, the chaperone exists, it allows for the misfolding of a whole bunch of other proteins, which for which the mutation to misfolding is rendered neutral or more nearly neutral as a result. So that's a pre-suppression kind of phenomenon. And it leads to increased functional dependence and by our definition, complexity. That's pretty well understood or documented in the case of chaperonins. I just Googled up this paper here. Most protein mutations and mutations that alter protein functions in particular undermine stability and are therefore deleterious. Chaperones, or the heat shock proteins, are often implicated in buffering mutations and could thus facilitate the acquisition of neutral genetic diversity and the rate of adaptation. And they conclude these results indicate that the protein stability is a major constraint in protein evolution, and buffering mechanisms such as chaperonins or chaperones are key in alleviating this constant constraint. So I would say that buffering, that they're using the word buffering to mean pre-suppression by my thinking. And so an example of the wrong thinking, at least I think it's wrong, it was published by Bill Martin and Eugene Koonin a couple of years ago now. They postulated that group 2 introns, which I will talk about in a minute, invaded the nucleus of eukaryotic cells and were so detrimental that the eukaryote, in response, invented the nuclear membrane and the whole nucleus, the whole nuclear apparatus, in order to avoid the onslaught of group 2 introns. I think that's definitely putting the cart before the horse. I mean, it would be far easier for the budding nucleus or the budding pre-eukaryote to simply avoid introns in some way. So, and I think it actually serves, when you think about it, Bill Martin's notion that mitochondria came at the beginning of the origin of eukaryotic cells, which I think is probably wrong anyway. So that's an example of the wrong way of thinking. I think the right way of thinking, which Tyler referred to, was the notion of the cellular bureaucracy. I think that many institutions and also many cells, particularly eukaryotic cells, are unnecessarily complicated, and this unnecessary complexity rises through CNE. That's what I think. And in particular, so then a good example of this would be the spliceosome. And I had intended initially to give this talk a lot about the spliceosome, but I think maybe many of the proteins involved in the splicing activity are exaptations, which is not so interesting to me. So I'll just go through it fairly quickly. So the spliceosome, there's approximately 10 or so introns per every gene in a eukaryote, so that's a ridiculous number. The whole spliceosome, which involves 5 small nuclear RNAs and about 100 proteins, has to be reassembled each time at each one of those introns and each one of those genes. Things which actually have the predecessors of these spliceosomal introns, namely group 2 introns, seem to do so much more efficiently, so that I would imagine that if we redesigned eukaryotes with only group 2 introns rather than spliceosomal introns, they would be much more efficiently operating, but what can you do? So so the spliceosomal origin from group 2 introns, the origin of the RNAs of a spliceosome, seems to be pretty well established now. The spliceosome, which is an intact RNA, it's actually a mobile element, so it involves a reverse transcription step. In prokaryotes, in particular in alpha proteobacteria, which were the ancestor of the nucleus, clearly is related to spliceosomal introns of eukaryotic cells, because there are structural similarities between each of the 5 snRNAs or at least most of them, and the group 2 introns in prokaryotes. So I'll just read this. Almost everyone believes, as I do, too, that Tom Kevler-Smith's 1991 hypothesis that eukaryotic spliceosomes arose through the acquisition by the last universal common ancestor of eukaryotes of the alpha proteobacterium that was to become the mitochondrion. Alpha proteobacteria have many group 2 introns as mobile elements, which are autonomous, self-contained, selfish DNAs in their genomes. They're infected with those things. Mostly not in protein coding genes, but sometimes. And the group 2 spliceosomal intron relationship was pointed out also in 1991 by Phil Sharp in his 5 Easy Pieces essay, which I just mentioned because it was such an obvious title at the time. And the intron encoded protein, IEP, on the top is clearly homologous to the most conserved spliceosomal scaffolding protein, PRP8, in almost all, well, probably all conserved in all spliceosomal systems in eukaryotes. So I think that's fairly strong evidence. So there was a paper by Julian Vosberg and Bernd Snell in Biology Direct about 5 years ago, or no, sorry, 8 years ago now, which I and Eugene Koonin and somebody else reviewed. So as you probably know, Biology Direct is where there's an intense interplay between the authors and the reviewers. So I probably badgered Vosberg and Snell into mentioning more about CNE, and then I realized that just last week that this was Vosberg's PhD thesis, and he just got his PhD. And he got his PhD by cobbling together 5 or so papers that he'd already written with Bernd Snell, who was a supervisor. So that's fine. And I'll just read what he says in the paper that I quoted from, nevertheless. Sorry. Numerous proteins were added to the spliceosomal core during eukaryogenesis. Many of these are clearly derived from proteins that already had an RNA binding function. Coincidental interactions with these proteins could have caused pre-suppression and subsequent dependence, increasing the complexity of the spliceosome without a clear benefit. The expanding repertoire of splicing regulatory proteins would have enabled the decay of clearly defined exon-intron boundary features, leading to dependence as well. In these ways, the present-day spliceosome would have been built up step by unselected step. And then later in the paper, he says, a neutral scenario in which these features were tolerated by a more complex spliceosome remains likely because a maladaptive intermediate stage, in other words, the cart before the horse stage, does not need to be invoked. Other advantages of spliceosomal introns and concomitantly the spliceosome, like enabling alternate splicing and fine-tuning, work on the long term. There's a strong feeling out there that the reason that we have spliceosomes and introns in eukaryotes is because they allow alternative splicing, and they've also initially that was supposed to allow exon shuffling too, the reassortment of modules of genes. I think those are both true, but they can't possibly have been the reason that introns were inserted in order that they might sometime in the remote future serve this role. So that they're very long-term potentials. So I think the spliceosome's subsequent evolution can be easily understood in terms of the falling apart of a group 2 intron into the 5 snRNAs, which are now functional in all eukaryotes. I think this is a pre-adaptation or a trans-level exaptation, depending upon whether you define exaptations as being things which arose at a pre-level, at another level, or not. So I think that Joe Belowski and I might differ in this respect. So most of the proteins, I think, probably arose by exaptation, although so certainly the proteins which have homologs in prokaryotes are clearly exapted proteins. Many of them are RNA binding proteins, in fact, so that they're predisposed to bind RNA. Those which don't have homologs in prokaryotes are possibly pre-adaptations, although it seems unlikely to me that a protein would evolve purely by chance, as it were. So many of the proteins are exaptations, whereas I think that the RNA itself is a pre-adaptation, depending upon whether or not we define things which evolved through selection pressure at a different level, because I think that group 2 introns are selfish DNAs, are pre-adaptations or give rise to pre-adaptations or to exaptations, and I think that's really up for grabs, really, at the moment. So how could it have happened? Well, I note that many spliceosomal proteins are already bound to RNA in prokaryotes. I mean, they were RNA binding proteins in prokaryotes. So we can imagine that the group 2 intron had a highly structured RNA. This fell apart in spliceosomal introns. But it already bound to a protein, which was the excess capacity or the pre-suppression activity, so that the structured RNA plus the binding protein existed already. This allowed for the RNA to relax some of its structural constraints, so less structured RNA plus now the protein becomes necessary rather than simply gratuitous, and that would ultimately lead to an even less structured RNA and even more necessary protein. So just to quote again from Vosburg's thesis, a neutral scenario in which these features were tolerated by a more complex spliceosome remains likely because a maladaptive intermediate stage does not need to be invoked. Other advantages of spliceosomal introns and concomitantly the spliceosome, like enabling alternate splicing and fine-tuning, work in the long term. So I think there are other features that have led to the spread of introns. One is the relatively recent discovery that I think coming first from fungi, but now shown to be more broadly distributed, of introners, or however you say that, which are transposable elements, so that sometimes the spread of introns within eukaryotes, particularly those which have a lot of introns, seem to be due to transposable elements embedded within the intron structure itself, or coterminous with the intron structure itself. And also, of course, Michael Lynch has frequently fixed on what, just read the letters here, genetic drift, fixation of mildly deleterious traits in small pre-eukaryotic populations. So Michael Lynch imagines that that large prokaryotic populations became small pre-eukaryotic populations, and therefore were more subject to drift, and therefore could more easily fix mildly deleterious traits like introns, for instance. And that's what happened. So I think that's probably true too, and probably led to the spread of introns. Okay, so just trying to rationalize me and Joe Belowski, because I think we might have a difference of opinion about this, and I'm not sure what Tyler's opinion is. If we assume that traits that arose through selection at one level did not arise through selection at another level, which they didn't necessarily do, and thus are preadaptations rather than acceptations at that second level, which is how I have carefully defined preadaptation as not being level-dependent selection, then at the organismal level, introns have a preaptive origin. So the statement that we made in that paper, thus the spliceosome may be largely a preadaptation for maintaining stable interactions with mRNA and carrying out splicing. However, it does not follow the vertex for pure preadaptation. The prior capacity of its proteins to bind RNA are acceptations, and it seems likely that at least some lineage-specific fine-tuning was accomplished by secondary adaptations. The important conclusion is that natural selection did not design the entire spliceosome, or necessarily even most of its components, for its current function. So there's a fly in the ointment, as there always probably is, which is, and actually Naomi Fest was one of my graduate students a long time ago, so I feel betrayed by her work, which shows rather clearly that in some eukaryotes, for instance, microsporidia, are here in terms of so whatever extremely streamophilic red algae, that introns can be lost. And you would think that CNE acquired traits could not easily be lost because it's a sort of a ratchet. And so I think that's still a mystery to how ratchet-like elements that are acquired by CNE, by the CNE ratchet, can nevertheless be dispensed with if selection pressures are strong enough. So that's my talk. And I will stop sharing so as to, well, if I can. There we go.
I guess we have time for two quick questions. Any questions in the room?
Comments in the chat.
Yeah, there was one comment by Steve, but I guess Steve was like an abstract of the paper, right? Steve, what is it that you put in the chat?
Oh, it's just a curiosity. I just learned about CNE a couple months ago, and I'm a big fan of the idea. I'm not critical at all. But what I previously knew historically was Lindquist's work, which was published originally in 1998.
Okay.
Started with something that where Doolittle mentioned about HSP90 as a buffer that allows protein misfolding mutants to be acquired because they're neutral in the presence of HSP90. And she built up a whole theory based on that, which developed into a field where many people developed the idea that they called evolutionary capacitance. And I think their idea is a bit different from CNE, and I was just musing about what the similarities and differences are. This is typical in any scientific field. Lots of people think of almost the same idea, but develop it along different paths. And I was just curious. Lindquist was a major figure in molecular biology and speculated a lot about evolution. Her speculations in evolution weren't always very good, but she was really quite brilliant in many ways. So I was just trying to understand the relations in what she was thinking and what people who use the CNE phrase are thinking they're similar but not the same.
Yeah, I think they're similar but not the same. I'm relatively unaware of Lindquist's work, and I probably should look at it. I think that the CNE idea is quite close to Bill Wimsatt's philosophy of biology notion of generative entrenchment too. I mean, I think the CNE concept has been reinvented still one more time by people who work on protein structure, and they call it entrenchment and contingency and contrenchment, I think, something like that. So
But the generative Pyler brought up this generative idea that Wimsatt yesterday. I've never heard of it before, but it struck me as a bit strange because it seemed like the idea was that earlier stages in development would be
sure
more constrained than later stages, but there seems to be overwhelming evidence that it's the middle stages that are constrained and the early and late stages that are free. And this is a widely discussed idea called the hourglass model or the phylotypic stage. And there's an immense literature on this in biology. And so I'm puzzled by why there's this philosophical notion of early constraint that's completely at odds with overwhelming discussion in the biological literature about the fact that it's the intermediate stages which are typically conserved. And there's a lot of reasons biologically to understand why that would be so. That's actually quite consistent with CNE. So I'm sort of puzzled by why you're focused on this philosophical idea that seems to be completely unsupported.
Well,
I'm just curious about these things. These are all new to me. I didn't know about CNE. I knew about these other things. So these aren't critical questions. These are just curiosities of how I can reconcile what you're saying with things that I previously knew.
Yeah, well, I don't normally read a lot of Bill Wimsatt, but I was just thinking that this is sort of a form of entrenchment. And his ideas about entrenchment, which I think speaks to the disconnect between philosophers of biology and the practice of biology often, says it speaks wonders, but so it's the idea of entrenchment, and I do think there was a temporal component to Bill Wimsatt's notion about entrenchment that isn't present in CNE, but
we have 1 minute, and Kerry has a related comment or question.
Yeah, I just think that I could help you, Steve, with your question about the difference between CNE and Sue Lindquist's work on the chaperones. So I think both start at the same place. They're saying, okay, the chaperones are a pre-suppressive mechanism, but then CNE focuses back on the chaperones and say, okay, well, now that you have these mutations accumulating, it makes you need the chaperones, and it focuses on the evolution of that. But Sue Lindquist's work goes the other way and focuses on what that does to evolvability. Like, now that mutations are cool, how does that change the way organisms evolve? And I think actually that's the area where some evolutionary biologists disagree with her. But I think both start at the same place. They just focus on different aspects. I hope that helps.
I think that's accurate. Yes, thank you.
Okay, let's think forward again. Thank you very much. And the next speaker is Alessandro Torri, who is here in person. Until recently, Alessandro was at the Pasteur Institute. He's still around the Pasteur Institute. There you are, kind of.
So, I'm afraid I stole a concept from Paul, so you will see. Be prepared, you will see the card again. Okay. Yes. So I prepared this work while I was doing my postdoc in Carla Sale lab at the Institut Pasteur, and it's a laboratory about insect immunology. And previously, I worked with antigenic variation in protist and previously in coagulation. And now I'm giving you a talk about evolution, the RNA interference. So I feel like I'm infiltrating an organization, and I hope you won't notice that. So let's introduce the RNA interference. The RNA interference is a complex molecular process that have both defensive and regulatory function. In animals,
transition.
So in animals, it's generally divided in three main pathways. The miRNA pathway, that is considered to be the regulatory pathway. The siRNA pathway, that's in insect and in plants, is considered to be the main antiviral immune system. And the piRNA pathway that exists only in animals, and it controls the transposable element proliferation in germinal cell. Of course, that all I just said is an oversimplification. The reality about RNA interference is more complex. So among these three pathways, the siRNA pathways is considered to be the more ancient.
Excuse me, we have a problem. The slides are not advancing on the Zoom, so there's a disconnection between what we're seeing here and what is seen on the screen. So I don't know why.
I have no idea.
So I guess you have to change the, do again the, stop sharing and do it again, please.
I don't see stop sharing in the.
Stop sharing, you see the red? Yeah.
Yeah, but I don't see it in the.
You escape from the full screen?
Yeah.
I mean, you can just do the same thing again. This is the power. Share.
Came out from the presentation now.
Do you want to go get out of it?
Yeah.
Just get back into it?
No, just the presentation. I mean, the full screen. Yeah. Presentation mode, you can switch off please.
I don't know how to do that because I never use PowerPoint. You'll have to do this. Sorry.
No, it's the same thing.
I don't know how to fix it.
On the screen, we see all the slides.
I think maybe Tyler is still sharing the screen and therefore
He's using my computer, so it can't do that.
Yeah.
Whereas Tyler could stop sharing the screen.
How do you see them?
Let's see this.
That's okay, right? It's not a bad thing.
Oh yeah, no, we see it now.
Okay.
That is in presentation mode.
Okay. Do you need the presentation?
No.
Can you, if you now swap this place?
I don't know how to do.
No, if you go back to the.
Yeah, but I don't see the command in the screen.
But you can do it by checking on that screen. You can see your arrow here or not? Okay.
So anyway, I can let if it's not a problem, I can let this presentation on.
Well, it's still not working now. I mean, that changed though. If you change slides, it doesn't work. I'm sorry for this.
Let's try to convert it to a PDF and then just run the PDF. So let's stop sharing this. Where's the PowerPoint? Save as. Yep. Let's save it as a PDF. Okay. And then, where did it go? Should be here. Search it. Should have somewhere. I might have just saved it in a weird place. So let's make sure I saved it on the desktop. Sorry. No, it's in the desktop.
At the top.
There we go. Okay, so let's try and get you to project this one. What you might do is that this is the one that's in PowerPoint PDF. How's that going now? Is it changing?
Yep.
Yep.
So, good start. So among these three pathways, the siRNA is considered to be the most ancient, and it was already present in the last eukaryotic common ancestor, LECA for friends, and this means that it originates in the ancestor of LECA. The main effector of the siRNA pathway are two proteins. One is dicer, is an RNase-free family protein that recognize double-stranded RNA and dies this double-stranded RNA, producing short double-stranded RNA. These short double-stranded RNA are recognized by Argonauts or EGO. EGO can load one of the strands and use this strand to target specific genomic region or specific RNA messenger. Now, as I say, the siRNA pathway is historically considered to be a defensive system, but in recent years, we see that its regulative function are more widespread than we thought, and at the same time, we discovered that the defensive function are less widespread of what we thought before. Sometimes it's difficult to disentangle these two functions. For example, in some species, the siRNA pathway produces the heterochromatin generation, and that allow the determination of the centromer. But this same process is the same that is inhibiting transposable element proliferation in the centromeric region of the chromosome. So it's very difficult to separate these two functions. Despite this, it's generally considered that the nature of ancestral RNA interference was primarily a form of defense against RNAi viruses and transposons. As far as I know, there is only one author that challenged this idea. He was Cavalier-Smith. He completely rejects this theory, and he said that the siRNA pathway probably evolved during eukaryogenesis to allow a proper chromosomal maintenance and chromosomal segregation in eukaryotes. Sadly, his hypothesis was in the very middle of a 60, 70 long pages paper, and it has been forgotten. Anyway, this is the main view about the origin of the RNA interference. It was a form of defense against transposon and RNAi virus, and this imply an adaptive evolutionary origin, as like eukaryotes are invaded by an incredible number of transposons and RNAi viruses. So the ancestor of the LECA had to evolve this new molecular system to defend himself. And this is problematic because, as Ford said, it's like putting the cart before the horse. Again, I stole that without shame. I'm not giving you the example about antibiotics, but the concept is simple. If the ancestor of LECA was susceptible to transposon invasion and RNAi viruses infection, it couldn't have the time to evolve a new molecular system to defend himself, especially in a world that was dominated by bacteria, which weren't susceptible to this invasion. Moreover, there is another problem, and is the RNA interference really essential against transposable elements? So we know that ectopic recombination and purifying selection are the main factors in controlling transposable element proliferation. We know that transposable elements are often self-regulated. This is especially true for prokaryotes, but genetic population studies demonstrate that it's possible also for eukaryotes, and we have evidence of that. We know that tolerance for transposable element can be selected even in the absence of RNA interference. This is the case of Drosophila melanogaster and P element. Drosophila can be selected tolerance for the P element without the intervention of the pi RNA pathway that is supposed to control this element. And finally, if it's true that LECA the ancestor of the LECA, unicellular organisms have low number of transposable elements because of they have a big population size and so have a very streamlined genome. They don't struggle with transposable element invasion, and the ancestor of the LECA was probably an unicellular organism, so should have been safe against this invasion. On the other hand, the RNA interference can reduce ectopic recombination and so reduce the transposable element deleterious effect. How can do that? With the formation of heterochromatin. The siRNA pathway can induce heterochromatin and can inhibit the transposons proliferation in centromeric region. Doing that, it increases the probability of fixation of transposable elements in the population. There is a biological example of that if we look at fungi of the Cryptococcus species. The organisms that still have a functional RNA interference have longer centromeres region with full-length retro element, while the organisms that lost the RNA interference have shorter centromer region, and they lost the full-length transposable element. So this leads to the hypothesis that in reality, the RNA interference can be the cause for the transposable element proliferation in eukaryotes rather than the defense against them. So if there wasn't this selective pressure for a new defensive system, how did the RNA interference originate? Well, prokaryotes have a very similar system that is called this antisense RNA regulative system. It's rely, I don't have a point. It's rely on this antisense small RNA with this complex secondary structure, and it can regulate the phage proliferation, it can regulate gene expression, it can regulate plasmid regulation. Basically, it's doing the same kind of activity that the RNA interference is doing in eukaryotes. However, these two systems, this prokaryote interference and the RNA interference in eukaryote, are not considered to be homologous because there is a lack of protein homology between the two systems. Prokaryotes have a protein called Argonaute, it's called P-EGO, but this protein do not participate in this process. So because of the lack of protein homology, it's generally the idea that the prokaryote interference is homologous to the RNA interference has been rejected. However, a lack of homology between proteins does not necessarily imply a lack of homology between two processes. This is called process homology. I give here an example with the binary fission. The prokaryotic cell division in alpha proteobacteria, the cell division rely, among a lot of other protein, rely on this protein called FTS. During evolution in mitochondria, that are organelles that evolved from alpha proteobacteria, you can see that mitochondria from some proteins like Dicotyledonum still retain FTS protein, but also use a new eukaryotic protein for the binary fission, it is DRP1, while in mitochondria from animal, plants, and fungi, they lost the use of FTS and only rely in DRP1. So during evolution, we can see this qualitative system drift. We changed the molecular machinery, but we didn't change the outcome of the process. That is still binary fission. So we propose that the origin of the RNA interference occur by qualitative system drift, a change in the molecular composition of the two processes, starting with the prokaryotic interference. How? And we propose two models for that. One about transcriptional silencing and gene expression levels, and another about translational silencing. So the first model, here you have a schematic about transcriptional silencing. It relies on this antisense RNA as I told you before, that is expressed by this gene in the chromosome, and it targets a specific RNA messenger. After the match, we have transcriptional silencing, and the RNA is free, degrade the complex, producing this short double-stranded RNA. These short double-stranded RNA are just junk. It's a byproduct of the reaction. They have no physiological function in the cell. Now, at this stage, mutation that inhibit, that lower the expression level of the antisense RNA are deleterious, because we need a certain level of this RNA for the system to work. So this mutation cannot accumulate, and they are erased by purifying selection. However, in the presence of Argonauts, EGO can potentially interact with this double-stranded RNA, short double-stranded RNA, load one strand, and use this strand to target the specific RNA messenger. This would cause an amplification of the interference signal, because now the interference is carried out by the antisense RNA, and there is this positive feedback caused by EGO that interact with the short double-stranded RNA. This could be deleterious for the organism, but molecular processes inside the cell do not act in isolation. They are all connected. So we speculate that if this happens, phenomenon of dosage compensation can fix the situation, reducing the expression level of the original antisense RNA. At this point, mutations that permanently reduce the expression level of this gene are no more dangerous and can start to accumulate in the system, and the system became irreversible and now rely on the presence of EGO to proper function. So from the prokaryotic interference to the RNA interference in eukaryote, we can see this neutral qualitative system drift, a change in the molecular composition of the system without any specific function being selected, nor the defensive function of the prokaryotic interference or the gene regulation function, but all the functions are maintained in the new system. The other example is similar. It's about the translational inhibition. These antisense RNA have secondary loops that can interact with the ribosome subunit, and this interaction interrupt the translation of a specific gene. After that occur, RNase E and RNase free degrade the complex, producing this short double-stranded RNA. Again, Argonaute can interact with this double-stranded short RNA and induce the amplification of the interference signal. Now, we have, in this case, transcriptional silencing. Now, PEGO, this excess capacity of PEGO made the translational silencing obsolete. So the system do not rely anymore in the transcriptional silencing. So mutations that can disrupt the loop, the secondary structure of this antisense RNA, are likely to appear. Now we have a linear or almost linear RNA, antisense RNA, but it can carry out the interference function in the same manner, because PEGO is taking care of the problem. So in conclusion, we propose that from prokaryote to the ancestor of the LECA, we have this qualitative system drift through constructive neutral evolution that allow the evolution of the RNA interference. And this is not the end of the story, because now eukaryotes have a system that can reduce the deleterious effect of transposons. And so we have the classic ratchet-like mechanism of constructive neutral evolution that lead to transposable element accumulation in eukaryotes, especially in metazoa. So the end, we don't need an adaptive hypothesis to explain the origin of the RNA interference. A neutral hypothesis is enough to explain all the effects produced by the RNA interference. And second, the RNA interference may be one of the main processes shaping the chromosome architecture of eukaryotes, biasing the genome evolution toward the insertion of transposon, pseudogenes, and other repetitive elements. Okay, I didn't have time to enter in the detail of our work. If you are interested, I suggest you to read the paper. There are a lot more detailed. It was published last year in PLOS Biology. And the original draft have a lot of examples about dinosaurs, because dinosaurs are cool, but we were forced to remove all that example. So anyway, this is life. And at the end, I want to thank the other author of the paper, Johannes Jaeger, Thomas Pradeu, and of course, my ex-boss, Carla Saleh, that made this project possible, and all the Saleh Lab for the discussion. And thank you. Thank you.
Okay, we have 5 minutes for questions.
I just have a brief comment. I really like that in the two models that you proposed, that they're both plausible together. And so you can have a case where a presuppressor is a presuppressor for two things at once. I mean, that makes it even more plausible that it'll get locked in as a presuppressor. The more things it's presuppressing right away, the more likely it is that the system is going to become dependent on at least one of those presuppressive effects. So I really like that about it.
Yeah, I'm sad we couldn't include the second image. So we have only to describe that in the text.
Yeah.
But yeah. Thank you.
Other questions or comments? Maybe on Zoom?
I think there are a lot of processes like this that where we're missing the how things evolved complexity, or how things changed over time, because we're thinking of the individual actors within each functional process rather than the process as a whole, and how the various actors could have changed over time. So I really like this example of that, and I think there's many other examples that could be used in the evolution of cells.
Indeed, in the paper of Nida Fedorov, she was the first one that suggested the RNA interference may be the cause of transposons proliferation instead of the cure. She takes care of all epigenetic mechanisms, not just the RNA interference. So there are different examples that may produce the same results.
Sorry, just to clarify there, do you think that in the other cases of mechanisms proposed by Fedorov, that they were cases where there was potential qualitative system drift? Or that they were other,
no,
so they're all potential presuppressors of T family expansion?
So that's complicated. We focus only in RNA interference here. Yeah. It's complicated because in a lot of species, epigenetic mechanisms like the formation of heterochromatin and the centromere determination depend on the siRNA pathway, but in other species, they do not depend on the siRNA pathway. They can occur independently of it. So generally, the species where the siRNA pathway is important are generally protist or basal metazoan. So it can be that we have another qualitative system drift from a system that was dependent on the siRNA pathway to a system that is independent from but is the same process, the formation of heterochromatin. And indeed, in animals, most of these tasks are carried out by the pi RNA pathway, not the siRNA pathway. So I think, yes, there is this qualitative system drift.
I don't see other questions or comments, so thank you again, Ali. We are now taking a short break of 10 minutes, and we're back at 1:15 for new exciting talks by Kerry and Jeremy, and then a general discussion, unfortunately without Olin, as I mentioned before. So see you in 10 minutes. Okay, Kerry, I think we're ready. So if you can share your screen, that would be great.
Yeah. Yep. Now I'm unmuted. Share screen. Just. You see a title slide, correct?
Yep.
All right. Okay, great. So I'm going to talk about the genotype, phenotype map. It'll be a slightly different talk from what we've heard before. I think because I've come to CNE a bit later and I've come to it from a different perspective. So it sounds like a lot of people come to it because they're interested in eukaryotic cells and how complex and or bizarre they are and how they got that way. I'm trained more as a population geneticist, and so I'm interested in sort of how to quantify CNE, I guess. And I thought about that a bit. I'm not really sure how to do it, but I at least think I know one of the steps is that you have to quantify complexity. You have to show that complexity has changed. I think there are other steps in terms of studying CNE, and maybe Jeremy will talk about some of those in his talk, or we can talk about those later. But I think there are these steps to show how complexity changes over time, show why complexity changes over time, maybe for neutral reasons. Maybe another way to study it is to just see what mutations do naturally to the level of complexity, so that you can see whether selection is pushing in a different direction. But what I'm sure of is, I think you have to define and quantify complexity in order to study it. And so that's one thing that I do in my research that I can talk about. I've thought about maybe different, simpler ways to quantify complexity. So I think it comes down often to counting. So I know you can count the number of genes in a genome or the number of duplicates in a gene family, the number of proteins in a macromolecule. It's too bad George isn't here, but I think that's what he works on. The number of genes in a pathway or the number of pathways in an organism. So I'm going to talk about something really weird, and it's counting the number of phenotypes in a cell. So I think a cell at some level has thousands or maybe an infinite number of phenotypes. You can imagine counting the level of every transcript. You can imagine counting the position or looking at the position of every nucleosome. There's tons and tons of phenotypes, but they're not all independent. We know a lot of them are related. So the transcription of many genes are actually related because they're all regulated by the same transcription factor. So how many independent phenotypes are there? And the question matters to me because I'm an evolutionary biologist and I want to measure fitness. So I want to understand whether the effects of nature or effects of a mutation are bad or good. Basically, I want to boil down all these phenotypes to, are you good and going to be selected for? Are you neutral or are you bad? I want to boil it down to something very simple. But to do that, I want to know how many inputs there are. Are there millions of different little phenotypes that can influence how a cell performs in an environment? Or are these things sort of all modular so that there's only a few main effectors, a few main levers that the cell can up or down regulate to influence how it performs in an environment? So this is kind of a strange and abstract question. So I'll start with an example of why I got into it. So I study yeast, and I was studying these two different yeast strains. One is from a wine barrel and one was isolated from an oak tree. And I noticed they look different. The wine cells are really round and the oak tree cells are more oval. The wine yeast have these occasionally these strange giant nuclei and the oak yeast don't. And so I wanted to do something easy and just map the genetic basis of those morphological differences. And I could do this because a lab had already created a QTL mapping family. So they mated the Y into the oak yeast, they generated 400 recombinant offspring, and they had genotyped that offspring at about 400 sites along their genomes. So what you can do then is pick a site on chromosome 1, sort these offspring by whether they inherited the wine parent allele or the oak parent allele, and then say, okay, do you have morphological differences? If so, it's probably that genetic site that's causing those differences. So you look for these correlations between genotype, whether you inherited the wine or the oak genotype, and phenotype, whether morphologically you look like the wine or the oak. So I did that, and here's a map of the results. So on the horizontal axis, you'll see genetic site, and on the vertical axis, you'll see morphological phenotype. There's 150 morphological phenotypes. I'm just showing you a few examples. And when you see a point on this plot, what that indicates is that there's a correlation there. So I think there's a gene on chromosome 8 that contributes to a difference in the bud radius of yeast. And there's a gene on chromosome 16 that contributes to a difference in the brightness of the nuclear stain. Here's where it gets interesting. So here's a gene on chromosome 13 that's pleiotropic. It affects 2 phenotypes. And here's all of my data. And what you can see is a ton of pleiotropy. There are these genes that affect upwards of 20 phenotypes. This one here affects upwards of 73 phenotypes. And we narrowed the effect of this particular QTL, which is a large region of the genome, down to a single gene called HOF1, and it affects 73 phenotypes. So that's very pleiotropic. And this was very weird because evolutionary biology, evolutionary theory tells us that this shouldn't happen, that you shouldn't have so much pleiotropy. The reason being that it causes selection to be inefficient. So if you have a mutation that affects tons and tons of traits, it's likely that some of those effects will be bad. And even though you'll be optimizing some traits, you'll always be ruining other traits, and you can never really get it right. It would be like if on your radio, if every time you turn the dial to control the volume, it also changed the bass, and it also changed the track that was playing. It'd be like every perturbation messes with so many things, and you can just never get the settings right. So the idea is that systems should have evolved to be more modular. In other words, to have a small number of phenotypes where the effects of mutation are sort of restricted to a module. But here we're seeing a lot of pleiotropy, and I didn't understand why. One reason why it could be some of the phenotypes I measure aren't really phenotypes that are independent. So I measure cell area and cell circumference, and that's just silly. Those are like 2 very related things. But there are examples of related phenotypes that are less obvious. So the reason tomatoes taste bad is that we selected for tomatoes that ripen to a nice uniform red color. And it so turns out that the mutation that affects color also affects taste. And that seems like a mutation that has 2 separate effects, but actually the mutation just affects chloroplast development, and that affects both color and taste. So there are a lot of related traits, and we don't exactly know which ones are related. We would need to know a lot of cell biology to figure that out. And I wanted to try and figure that out because I wanted to understand why I see so much pleiotropy. Am I just sort of observing tons and tons of related traits, or am I actually seeing mutations with effects on independent traits? So to figure that out, I did some single cell work in yeast. I took yeast clones, which are just a yeast strain divides asexually, it makes a lot of copies of itself, and then you get a bunch of different cells. Even though those cells are all genetically identical, they're not phenotypically identical. They have morphological differences because they're in different stages of the cell cycle, or they had unequal access to nutrients, even though they're in the same tiny little well, or they have differences in their age, or just stochastic differences in gene expression. So what we can do is look for correlations between morphological phenotypes within clonal cells. So you'd expect phenotypes like cell area and cell circumference to just be strongly correlated no matter what, because those traits are very similar. But maybe other traits aren't correlated. So we asked, are most of the traits we measure inherently correlated? The answer is actually no. So for every pair of traits, every dot in this plot here is a pair of traits. And you can see the median pairwise correlation is pretty low. But then if we ask, do these genes tend to influence correlated traits? The answer is yes. So here's a pair of traits on chromosome 2. There's just a single pair of traits affected by this particular QTL. and they're very strongly correlated. Here's another QTL on chromosome 2. This one affects many more pairs of traits, but on average, the median correlation is pretty high, higher than what you would expect if you just sampled from the null distribution. And so by and large, the genes that I study seem to be affecting related traits. So a lot of the pleiotropy I see is what you would expect. It's because these traits are related and not because genes are affecting lots of independent modules. After we did this analysis, we realized that we can actually use these correlations to kind of paint a picture of the genotype, phenotype, phenotype, phenotype map. We can look at modularity. And we can do that by creating a force-directed network where every node is one of the traits we study, and every edge represents the strength of the correlation between traits. When we make these kind of networks, you can see these modules form. So here's one, here's one, here's one. You can actually ask if this network is more modular than you would expect by chance by just perturbing which edge goes with which nodes. And we do that over and over. We see that indeed this network on the left is more modular than all of these other permuted networks. And so there's some evidence there that biological systems did evolve to become somewhat modular. We can also see that genetic changes tend to affect modules. And here's just a little picture of it, but this tends to be true. You don't see genetic changes affect random little dots here, but you can see that their effects tend to be constrained to modules. And so that's one way to study the genotype, phenotype, phenotype, phenotype map, to actually measure phenotypes and to see how modular things are. I don't know to what extent this helps us think about CNE, but I thought it was one way to study complexity that might be different from what we're used to thinking about. And I'll talk about one other way. Yeah, so this is kind of an abstract concept, and the next one is even more abstract. So this way to study the number of phenotypes affected, you don't even have to measure phenotypes. In fact, you don't have to know anything about phenotypes. The idea is that you take a bunch of mutant strains and you measure their relative growth rates in a whole bunch of different environments. And if you find a strain that has a different gene by environment profile, you could also call this a growth profile, you can infer it's affecting different phenotypes because those phenotypes that are affected are going to dictate how it behaves in these environments. So if it's behaving differently in some environments than these other strains, you can say, all right, it's probably affecting different things at the molecular level or at the cell level. So the conceptual leap I want you to make is that if we build these types of profiles for a lot of mutants and then cluster by the type of the profile we see, we can infer sort of the minimum number of phenotypes that must be affected by all the mutants. So in this example here, at minimum, 2 phenotypes must be affected. The red and the blue affect one, and the green affects a different one. I can study this in high throughput because I study barcoded yeast strains. So I can make a whole bunch of mutants, barcode them, and then mix them all together in a single flask. And then I can study their different fitness and environments by how these barcode frequencies change over time. And I can do this, I don't know, in 2 weeks for hundreds and hundreds of mutants. And I can also do it in 2 weeks for up to 50 environments. So you can see here some of the strains grow faster in some environments than others. And that's what I use to generate these growth profiles. Let me show you some of the profiles that I've generated. So this is a growth profile for mutants in a gene called IRA1. This is a gene that participates in a glucose regulatory pathway, and the mutants that I'm finding in this gene all basically shut it down. They're frame shifts or stop codons. Some of them are missense. The way that these yeasts or these mutants were found is these yeasts were evolved under glucose limitation, and they're given more glucose every 48 hours like clockwork. And so the idea is they don't really need to sense glucose anymore. They're just getting it every 48 hours. They don't need to bother turning on or off their growth machinery. If they wait it out, they're going to get sugar. And that's really beneficial in environments that are glucose limited. But in environments that are very different, like high salt environments, that's not too beneficial. It's not good to just turn off your sensing machinery. So here's another growth profile for another mutant called PDE2. You can see there's some similarities to the other growth profile, but there's a difference here. In these 2 particular environments, this mutant is really advantageous. And we didn't see that before, but we do see it for another type of mutant called GPB2. And these mutants are all regulators of the same glucose sensing pathway. So now the question becomes, okay, I have all these growth profiles. How do I cluster them to figure out how many phenotypes are affected? I do that using singular value decomposition. Basically, what it does is it infers the number of phenotypes that must be collectively affected by all of these mutants in order to explain the variation in the growth profiles we see. So it infers how many phenotypes there are. I'm going to call these phenotypic components because this is weird, right? They're not really phenotypes. They're not things I measured. They represent causal effects of each mutant on fitness. So they're kind of like phenotypes. Singular value decomposition also infers the importance of each phenotype in each environment. Okay, so now the big question, how many phenotypes underlie all the growth profiles? I guess I should have known that the answer to the ultimate question of life and everything is 42. Just kidding, it's actually 8. But I think there is a funny parallel here. So if you didn't get the joke, it's from Hitchhiker's Guide to the Galaxy. And in that book, these mice ask, what's the answer to the ultimate question of life, the universe, and everything? And it's 42. And that answer kind of falls flat. Like, what does that mean? And so we similarly ask this big question. The answer is 8. Okay, what does that mean? Let me show you what we can do with these 8 phenotypic components. So we can make predictions of how mutants behave in held out environments. So if we just use one phenotypic component, a one phenotype model, we get terrible predictions. Some of these predictions are 0% accurate. But if you use an 8 phenotype model, you can see we get much better predictions. Let me hone in on one to help. So in this particular environment, a 1 phenotype model or a 4 phenotype model make pretty terrible predictions. You can see GPB2 mutants here are predicted to have the same fitness as PDE2 mutants. See, they have the same level on the vertical axis, and that's just not true. But an 8 phenotype model makes better predictions. So PDE and GPB2 are now predicted to have different fitnesses. And so what this has started to tell us is that these 8 phenotypes aren't meaningless things, that we've actually captured something meaningful about biology that we can use to make predictions. But I think it's still kind of unclear what these 8 phenotypes are. So let me try to explain it one more time. So our model outputs hundreds of genotype, phenotype maps that trace how different mutations affect 8 different growth-related phenotypic components, and how each of these phenotypes contributes to growth in a given environment. And we use these to make predictions. For example, from the thickness of the arrows, you can see that one of these mutants has a stronger effect on the 8th phenotype than the other. And this phenotype might not contribute to growth so much in environment A, but in other environments, this 8th phenotype really matters. So since GPB and PDE mutants have different effects in this phenotype, our model predicts their different growth rates in this environment. And this is cool because GPB2 and PDE2 are negative regulators of the same glucose pathway. Like I would have figured mutants to this gene have the exact same effect in every environment, but our model's saying that they don't. And so that's teaching us something kind of cool about cell biology. And it's saying that they're doing something different to cells. More than that, it's telling us how we can figure out what they're doing, what those phenotypes are. Like the next logical step would probably be to study these mutants in this environment and to try and see if we can figure out what that phenotype is. So even though we're identifying these really abstract phenotypic components, our goal is to try and figure out what's happening at the molecular level. So I don't know if this helps us think about CNE, but it's another way to think about complexity and to quantify complexity in this sort of high-level way, a very high-level way. What are the abstract phenotypes in a cell that matter to fitness? And I don't know, maybe that will provide some food for thought for our next discussion. That's it. Thanks.
Thank you very much. We have time for questions, comments.
I have a question. Oh, go ahead.
No, please.
I have a question which I think I can state quickly. So in the second part of the talk, is it the case that that number 8 falls out approximately as an assessment of your best assumption of the amount of pleiotropy that it takes to explain the difference in growth rates? Is that sort of what that number represents? So it's sort of like assuming that that the given gene has 8 pleiotropic effects helps you best explain the growth rates.
Yeah, I think so. So the word pleiotropy is kind of tough because in reality, I think these mutants have a bazillion pleiotropic effects. I think they affect a lot of these little tiny things, like the level of this transcript, I don't know, the level of this metabolite. And so we're saying, okay, there's these different concepts of pleiotropy. One is called true pleiotropy. So we're saying, okay, how many real independent things does it affect? And the answer to that question is 8. There's 8 different things that matter to fitness or to growth rates in these environments. Is that what you're asking?
Yeah, sort of. If it's approximately a measure of optimal pleiotropy assumption. So it's sort of like, it's not individual pleiotropies that you're measuring. You're not saying there are 8 of them. You're saying something like, if I assume that there are 8 major key underlying pleiotropic effects, then this best explains my growth rates.
Yeah, that's right. And it could have not worked. And I think that's why we were happy. It could have been like, there are a bazillion different things that are affected, and you can't boil it down to a number like 8 or even a number like 15. There's just too many different growth profiles, and there's no way to capture that. So it was nice that it boiled down to a small number. And I guess I wonder in other systems it would be the same. So we're trying to repeat it for other mutants that affect drug resistance. I wonder if you repeated it for random mutants that had nothing in common, or if you repeated it for other organisms or mutation accumulation lines. Like, would you suddenly find, okay, these weird mutations are affecting lots of things. They're not falling out so that they only affect 8 main regulators of growth. And then maybe you would see more complexity. I wonder what dictates how much complexity we see. But yeah, I think what you're saying is right.
Yeah, I would wonder if I can also make one comment. I would also wonder if you would see more of them if you had less modularity in the underlying structure of pleiotropy. So if there was bigger modules, you'd see less sort of underlying pleiotropic effects that were sort of major and independent from one another. But if you had many tiny modules, maybe you'd see more of them.
I think that's the idea. Yeah.
Yeah. Kerry, you wanted to add something?
No, I was just going to call on the next person, but you probably should do that.
Well, yeah, because we have some people here that probably you cannot see. So yeah, it's okay.
I would like to go back to it's going to be a very naive question, going back to the initial argument where you said that to think about CNE, one important thing is to have a measure of complexity. And I missed something about the initial argument about counting phenotypes and in which way an organism that will have evolved more phenotypes would be more complex. I'm sorry, I missed the initial argument about how counting phenotypes or having more phenotypes would be a measure of complexity.
Yeah, I guess I don't know what I would consider complexity. So I think I'd be interested in discussing it. So yeah, I don't know. In some way, you could argue that it would be far more complex in an annoying sense if there are many, many independent things. Then it'd be very difficult to predict how mutations affect fitness because you'd have to understand all those things. So that would be one definition of complexity. Modularity, in a sense, would then be less complex. But I don't know.
The definition complex being annoying. From a practical point of view, it's very useful.
Okay, Ford.
It's not so hard to distinguish relative complexity, though. I mean, if something is more complex than another thing, you know, like if you add an extra component to something that already has a gazillion components, then there's a gazillion plus 1 components, then it's more complex, right? Sort of, but that's not my question. You're measuring fitness effects, right, of phenotypes, not just phenotype alone, but you're measuring fitness effects, I think, by the barcoding method that you use.
Yeah, that's correct.
Yeah. So the phenotype might actually be quite variable, but some of it doesn't matter.
Well, I think the idea is that I'm not sure what you mean. We're inferring the phenotype. The phenotype is this abstract causal effect of a mutation on fitness, but I have no idea what it is, and it could be like a nonlinear combination of phenotypes.
There could be more phenotypes than there is fitness affecting phenotypes, I guess. But yeah,
yes, yes, it can affect many phenotypes that don't affect fitness.
Right.
And the hope is that because we study 50 environments, we'll catch them all because it'll matter in some environment, but maybe we didn't sample enough environments, or maybe you can change something about a cell that, and I think maybe you would all agree with this, that actually has no fitness effect, and we would never see that phenotype. You have to affect fitness for us to catch you.
One last question by Alok.
Hi. So all the discussion about complexity, I want to ask if you ever try to think with Kaufman and Wimsatt from the 70s, when Wimsatt distinguished the descriptive complexity, interactive complexity, and I think he's missing a third. And it all came from Kaufman's articulation of parts explanations in biology. And it feels like you could build some kind of ontology and ability to make quantitative metrics of complexity from those two plus a third. This is just a question if that literature has ever shown up in your horizons.
I guess not, but it'd be cool to look into it. I know more the perspectives from like Gunther Wagner, who argues strongly for modularity, and that
So
kind of
the
debate
key.
there.
I mean, so what was very nice about your trying to build a way to do natural history of complexity, right? Your phenotype is an articulation of let's do the natural history of complexity inside the system. And I guess, and pleiotropy puts a crisis on the whole modularity thing, because the same module participates in very different wholes or subsystems. And how do you, I mean, so when Ford was just talking about if you go from n to n+1, you've got some increase in relative complexity. Well, if you don't go from n to n+1, but you add, but there is the number of systems being represented in that n increases in m to n+1 without increase in number of parts. How do you describe that? And so anyway, so this is very exciting, and thank you, and I'll write to you separately.
Yeah, no, that would be cool. It would be like mutations tried to increase complexity, but the cell said no. Like I was still sort of canalized into these things that matter, something like that.
Biochemical pleiotropy of chaperones, same components, same limitations, but many more biochemical activities. I mean, that's another example. So thank you.
Thanks.
Well, that's how we categorize the increasing dependency relationships, even though the same number of components. I mean, that might be C&E.
Yeah, that's why I generally define it so that it involves increases in essential complexity or dependency relationships rather than overall net complexity or something like that.
Yep.
But yeah, both of them happen.
Okay, let's thank Kerry again. Thank you. And now the final talk by Jeremy, Lynchian considerations. It's not David Lynch.
Lynchian is a great noun or adjective for someone's name. That's a good one.
Yeah, I've enjoyed. I don't. Mike doesn't actually know I'm giving this talk at all, so I think that's also funny. Hide floating meeting controls. Wonderful. Okay, I can see some of you. You guys can see my screen. Yes. Wonderful. I really like being able to see as many humans as possible on my screen, because then I know when people laugh at my jokes. Highly mutually reliable. Okay, so Lynchian considerations. First, I want to say I was really excited when I first got the invitation for this meeting. I spent my honeymoon 10 years ago in Bordeaux, and I was very excited to visit again. But this will have to do for now. I'm hoping to get there again. But today's talk, I will get around to talking about some Lynchian considerations at the end, and I'm really hoping I can generate some discussion, because I'm not a population geneticist at all. But I work down the hall from Kerry and Mike Lynch, and I've been, I think, overly influenced by their proximity. So Mike, as many of you may know, he's understanding of the CNE cause, but every time I talk to him about it, he seems quite frustrated by the verbal nature of CNE as a theory, and the lack of mathematics. So I want to kind of take this opportunity to amplify that concern with a room or a Zoom room full of people that also care about CNE. Although, since I'm neither a mathematician nor a population geneticist, I can't necessarily deal with many of Mike's potential concerns with CNE as a theory. So I'll kind of reiterate a few of the things that have been said today. I'll talk about a little bit about my own grappling, my own kind of struggle with CNE in the last couple of years, and then I'll kind of try to open up the floor to thinking about CNE from a population genetic perspective. Okay, so I've ever since this paper with at least a couple of people familiar to many in the room, including Ford, who's in the room, I've thought of adaptation as not as a null hypothesis, and I've thought of neutral explanations and CNE as potentially a null hypothesis that we need to disprove. So I kind of fell in love with CNE explanations, and at least on my own time may have over applied CNE, but I think maybe as this story will make clear, that it can be useful to take CNE as a hypothesis that you need to prove wrong in some situations. Okay, so we know what CNE can explain quite comfortably. And so this is an example that I was reminded about yesterday in one of John McCutcheon's students' committee meeting, where there are these bacteria where in the ancestor, the endosymbiotic bacterium Hodgkinia has a single genome with about 165 genes encoded. But then there are lineages of these Hodgkinia that live inside cicadas where the genomes have fragmented. There's no gain of genes in these genomes, and some of these genomes only have one essential gene, but they've fragmented to ridiculous extents. So I'm very comfortable with this being explained by CNE. There's no adaptive explanation I can think of where you would want this kind of chaotic complexity evolving. And many of the genomes here actually exhibit some of the problems that Anthony described in the Buchnera example earlier. I think we're comfortable with some of Arlen's original examples where CNE can explain genomic rearrangements. I'm not comfortable with any adaptive explanation for the micronuclear, macronuclear genome rearrangements that occur in ciliates. The other example that's commonly mentioned is the RNA editing of kinetoplastid genomes. However, I've been noticing that there are actually several genes that are not edited in kinetoplastid mitochondrial genomes. And I've been wondering if there are actually mechanisms of where pan editing can revert to non-edited forms, or perhaps there is some reason that the ratchet stopped moving forward in certain lineages. And now the other kind of molecular complexity that CNE can explain is the expansion of complexes. So these are some fairly recent examples. This figures from this review, but the example is from a Thornton paper looking at the expansion of the vacuolar ATPase components in fungi. So in this example, the ancestral vacuolar ATP synthase of eukaryotes has one extra subunit. For some reason, there was a duplication and it was retained. And then in fungi, this seemed to have happened again. There seems to be no good reason to add this component. And they did experiments to show that an ancestral version of this complex is approximately as fit as the extant versions. In a similar vein, the Thornton lab has reconstructed ancestral states of hemoglobin, and they show with I think quite convincingly that this first step of dimerization likely had no obvious benefit to the so this dimerization could have been neutral, but then they show that the tetrameric form immediately gains these emergent properties that are adaptive. So in this increase in complexity, it appears that there's a stage of increasing complexity that was neutral, but then there were immediate gains, phenotypic gains, adaptive gains upon tetrameric changes, or the gain of tetramerization. And what's really important here is the Thornton lab was, I mean, it's just a few mutations. It's a very small number of mutations that are required to go from monomer to dimer to tetramer. And then George, who's unfortunately not here, who gives an excellent talk on this subject, simplifies his papers incredibly well, such that I can't actually explain this near as well as him. But through a similar set of experiments, through recreating ancestral-like forms of rubisco, he shows that this increase in complexity pretty much instantly provides an adaptive benefit. So showing that in this case there isn't a non-adaptive, there isn't a neutral explanation for this increase in complexity. And so now turning to something I've been thinking about for I mean, in one way or another, 20 years actually. ATP synthase has always been on my mind, and I've recently done a comparative genomics project. So in the last few years, several ATP synthase structures have been solved from various lineages. And our kind of canonical ATP synthase found in fungi and animals has only 17 subunits. But there are a bunch of other lineages that have a whole bunch of extra subunits, and none of these subunits none of these accreted subunits are actually related to one another. They're completely independent expansions. And so I was curious if these could be explained by constructive neutral evolution. And it's important to note that each of these variations in ATP synthase structure correlates with differences in mitochondrial cristae structure. So mitochondrial cristae are the invaginations of the mitochondrial inner membrane where protons accumulate that power the ATP synthase to make ATP. So you can conceive that the variations in cristae structure could change the biophysics of the proton motive force, and could these different shapes could very well be adaptive in nature. So the first question was, are these 17 subunits that are present in animals and fungi, are they actually representative of some of the ancestral ATP synthase? So a volunteer high school student in my lab did this work, and he was able to show that all 17 subunits that are present in animals and fungi are present across all eukaryotes. So we can conclude that yes, this 17-member subunit is probably representative of the last eukaryote common ancestor. And he was also able to show that the changes that occurred in ATP synthase to make these weird structures, they occurred billions of years ago, and they seem to occur at the base of these major lineages. So ciliates have a strange ATP synthase. The mysozoans, which include dinoflagellates and apicomplexans, have a strange ATP synthase. The chlamydomonad green algae have a strange ATP synthase. And then both the trypanosomes, the relatives of trypanosomes and the relatives of euglenids have strange ATP synthases. And it's important to note that based on the most recent estimates, all of these changes occurred over 500 million years ago, maybe 1 billion years ago. And there has not been that much change that we know of in these complexes since. So all of the various extra components seem to come all at once, and they've been retained in most representative species in each of these lineages since they came on. So that means either A, all of this complexity happened kind of all at once very quickly, or these changes occurred over a long span of time that was completely erased by extinctions and similar events. And so I was originally quite somewhat convinced that ATP synthase has a singular simple function of making of making ATP for the organism, and now I'm less convinced by that, and I think there are these other phenotypes larger scale phenotypes that may be felt by selection. So changes in ATP synthase structure, changing the morphology of cristae, changing changing the proton motive force, and all of these could be related to small changes in the ATP requirements of a given organism. That doesn't mean CNE can't explain this, but we have to think about this iterative complexity that's building up in these various lineages, and think about the kinds of population genetic situations where this could actually happen. So I think we're comfortable with CNE being plausible in the case of increasing from a monomer to a dimer, or changing a homodimer into a heterodimer, and I think this has been thought out fairly well. But how might we get this larger kind of molecular complexity that we're seeing in ATP synthases neutrally? And I'm not sure if it's quite possible. And so something that I have not seen in the CNE literature yet is how long in terms of generation times would it take to entrench like one extra subunit? How long would it take to accrete 3 subunits, 10 subunits? What is the reasonable timeline for this kind of accretion? So, stealing from one of Ford's papers, we have this population genetics. We have this neutral ratchet of complexity. And so I want to try to think about the population genetics behind this neutral ratchet. And so I think just to simplify things, we need to think about all of these steps as reversible, which is kind of indicated here. This is the reversion of this, just shown as a different path. But I think it's easier to think of each as two-way arrows, and between each stage, the probability of going backwards should be the same as going forwards. So I think, and Kerry, I'm hoping you'll correct me whenever I'm wrong about anything here. So feel free to chime in whenever. But we have, so we need to think about mutation rates, fixation rates, and we need to think about fitness as well. So in CNE, the assumption is everything is neutral or very nearly neutral, and I think I keep reiterating this because I was fascinated by it when I found this out. But population geneticists consider fitness difference of 10 to the minus 5 to be rather astronomically large. And so a fitness defect of 10 to the minus 5 would be wiped out of most even medium-sized populations. So 10 to the minus 5 is a huge fitness difference, and cell biologists like myself have never imagined measuring that small of a fitness difference, or even necessarily finding it interesting. So that means these changes, this excess capacity represented by the ability of B to bind A can have absolutely not much of a fitness defect. So now we have a second mutation. So these pre-existing suppressing elements have to fix in the population and be, or it could just be one of these excess capacities that already persists in a population, such that a mutation, which if these suppressing mutations were pre-suppressing mutations were not already fixed in the population, any individual that gains this context-dependent deleterious mutation would be wiped out by selection. So in a population that has these yellow mutations fixed, the red mutation will be allowable. And then we can see how this could lead to some extreme functional dependence. But my question is, how long would we expect this to take? All these neutral mutations, or potentially very nearly neutral mutations. How long would they take to fix in the population? And then how long would it take to fix the next, the C subunit, the D subunit, et cetera, et cetera. And so I haven't thought about modeling this I haven't thought too deeply about modeling this. I have been thinking about how neutral endosymbiont gene transfer could occur. So I don't want to get into this, but I think there are somewhat similar models would be required. But so I guess I'm curious what you all would think are the necessary parameters that we'd have to model for this iterative increase in complexity. And what would this model even look like? What questions could be answered beyond how long would it take? What mutation rates are needed? What population sizes might be more or less realistic for this to occur in? And under what situations could at least aspects of our current understanding of constructive neutral evolution be proven wrong? So I'm just going to stop there and try to get some discussion going. I want to thank Savar, who did all of the ATP synthase stuff, and point out he's just finishing his first year of undergrad now, but he did all of the work as a high school student, and he'll have 4 authorships before he turns 18, which I'm jealous of. Okay, questions, discussion.
Thank you very much.
I'm going to make it a habit of just offering my comment at the end, if that's okay. Yeah, so I think, okay, so I remember talking to Mike about this a little bit, but I think there's something wrong with the model that you have where mu is the same for all transitions. So I think it's okay to think that the first, unless there's, so the first step, so the acquisition of B, that might have, both directions of that might be equal probable, but all the remaining steps are not equal probable. I think you're more likely to mutate from the second stage onto the third and so on than you are to mutate back. And that's because of an artifact of this model, which it seems like individual mutants sites in B are pairing with specific individual sites in A. But in fact, what's more likely is that there are many potential sites in A that could be changed, that can be pre-suppressed by the very same sites in B. So for instance, there's many different misfoldings of a protein that can be suppressed by the same chaperone than there are ways to regenerate the same folding pattern. So in those cases, you're more likely to acquire misfoldings, and there are many more misfoldings to acquire than you are to reacquire proper folding without chaperones. So I'd say the model has to have an asymmetry going to the right, and that's just the ratchet element that's embodied in CNE.
I agree that there are more mutations available, but you still have to have fixation of those mutations, of each one of those mutations, or you have to have recombination that ends up resulting in more of these individual mutations being fixed in one in one population, right? So you have to think of I mean, it's not like one mutation occurs and then another one and another one in a single mutation accumulation experiment where you have like single cell bottlenecks or something. You're dealing with a likely breeding population. So does that make sense? You're still
so in sexual populations, it could be the case that you have many different types of misfolding mutations occur in different subparts of that population, and each one of them drifts closer to fixation or something like that. So it's not like you're fixing one at a time. Yeah, so but I think even that would in general speak again in favor of an asymmetry in the model, where if you're just thinking about the overall likelihood that you'll increase dependence versus decreasing dependence, that is, move to the right versus moving to the left, then the possibility for multiple potentially different and independent folding mutations to emerge within a population would speak in favor of it moving closer to the right.
Maybe we'd have to model it, I think.
Yeah.
I mean, I agree that there would be more, I mean, there's more chances of these various mutations accumulating, but I don't see how that necessarily relates to fixation. I think Ford was first, but Kerry, are you going to add directly to this?
No, Ford can go ahead.
Okay.
Your boss, Michael Lynch, published a paper in, I think, 2015 about the frailty of the adaptive hypothesis, and he articulated the notion that the earliest eukaryotes were very small populations, so that fixation of neutral mutations might have, I mean, deleterious mutations might easily happen. And I actually had this discussion in the chat with Jeremy Poole, that's Anthony Poole, previously, and yes, I mean, it, you know, just as the frequency of mutations decreases with decreasing population size, so the number of actual mutations, neutral mutations, decreases as the population size gets smaller, but the actual effect of the selection on those mutations gets smaller too. So neutral, the slightly deleterious mutations, unlike what you said at the beginning, couldn't be fixed in small populations, and so maybe it's in eukaryotes. That's, you know, that's where all this cellular complexity comes from.
Yeah, and I guess, I mean, what I'm trying to point out here is that by modeling it, we can answer the question of how small did those populations need to be, how long might it have taken, and how deleterious could the mutations have been.
Yeah, no, but the other thing that Tyler did point out was that the deleterious mutations can happen all over the board, whereas, you know, it's not just a single one like this picture shows.
It can happen all over the board, but what's unclear to me is how each of those would fix.
Well, the more there are, the more likely they are to be fixed. That's all.
One of them.
More likely it is that any given one of them will be fixed. So in the
Yeah.
picture, it kind of looks like you have to pair up the red dots in A with the yellow dots in B. But in fact, what you can do is just pair up any number of different potential red dots in A with something in B.
Right? Yeah. Even just the fact that there's this, and I think it could be in some of George's papers, or it could just be that George is the one that explained it to me well. But once you have this interface, you can have any number of mutations forming, and there's a bias towards hydrophobic residues, and that's all. I completely accept that. It's just a question of how long does it take to fix each mutation, or does it actually, or once you have a couple mutations, does it actually tend towards some kind of adaptive stage, and then a new a new iteration of CNE occurs, and then there's an adaptive stage where you're honing the ability of the complex to actually be stable. Kerry.
I guess one thing I wonder about excess capacities, like in the chaperone example. So the cell does have an excess capacity. There's more chaperones than it needs. And so if you have a mutation that creates a new client, that's okay, because that new client will get folded and then it will get locked in. It will become a new client, and you can keep adding new clients. The ratchet can keep ticking. And then very quickly, doesn't the excess capacity run out? And so that's clearly not what happens. Cells still seem to have an excess capacity. And one argument, yeah. I'll go ahead for it.
Would be jammed up with the clients?
Say again.
You mean that the chaperone would be just, you know, clients would be waiting in line for the services of the chaperone?
Well, what you see is that at some point the cell, and it's pretty early, the cell says, okay, I need to up expression of my chaperones. If you increase the temperature, or if you add a misfolded protein to the cells, like if you engineer it, they don't have an excess capacity as is. They need to upregulate the chaperones. And so is that what happens?
Well, that paper that of yeast in yeast that or maybe it wasn't yeast, but no, it was E. coli actually, but where they overexpressed the chaperone gene and they resulted in increased frequency and mutations in the clients of the chaperone, which is interesting.
And so does it always get to this same breaking point? Like at some point, selection has to kick in and say,
Sure,
you know, there's no more excess capacity here. And why aren't we already at that point? Like if mutations always happen and new clients are always being added, why aren't we always at the breaking point where there's no more room? Shouldn't we have very quickly gotten to that point?
Well, we could be kind of at that point, but then you kind of have the turnover that sorry, I've forgotten names, and I don't have all of the names here. In the awesome presentation about the process homology, so the chaperone problem, you can think of that as like process homology, whereby I mean, if you have something new coming in, a new client coming in, maybe you have a client going out. So there's not necessarily.
It's also important, I think, to distinguish two types of excess capacity that are going on in the chaperone case. So one is the excess capacity in terms of the actual excess of the quantity of chaperones relative to the folding assistance that's needed at a given time. And the other is just the mere existence of chaperones at all that can sometimes, when they're not folding the proteins they need to fold, potentially fold other new mutants. That's also an excess capacity. So there's sort of an excess in quantity of the chaperones, but there's just the mere existence of them is also an excess capacity. They're promiscuous. Well, they have to be. Yeah.
Yeah, specialized to help one specific.
Yeah, that they can help with lots of folding. Yeah.
Alok, you have a question?
I have a small question, and then what might be a mid-sized comment about the conversation about chaperones. Can I ask which biochemical example did you make this diagram out of? Maybe you mentioned
this is from one of Ford's older papers. I don't think that there's any specific biochemical example. I think this is just a general model for how you can go from a monomer to a dimer and require that dimer with no increase in function.
So let me hang a biochemical example on this, right? So just for the kind of my comments, I got trained in protein biophysics. I worked on the protein folding processes and protein interaction stuff. So this is in some level the world in which I found my scientific head. So monomeric myoglobin ends up becoming this complex tetrameric hemoglobin and enables a lot of interesting metazoan biology. And you could put that on this diagram and say, okay, let's learn something from it. What caught my eye was you talking about that the protein-protein interface has a lot of hydrophobic contact, which tends to be non-specific. And yeah, it's a problem for developing drugs that inhibit protein-protein interaction, because there is, you can call it tolerance, but it's exactly the same tolerance that makes chaperones so powerful. And now I'm going to switch into the conversation around chaperones and the buffer capacity of chaperones and why do you upregulate chaperones. So maybe it'll help to use a concept that uses complexity in a negative way, right? Let's say the cell has an enormous amount of negative, destructive complexity. The crowded cell soup can become an irreversible omelet at any time. And what the chaperones are doing is blocking that. And so they are managing destructive complexity. They are the cell's resource to manage destructive complexity. And that's why HSP, heat shock protein 90 or heat shock protein 66, right, all first discovered in prokaryotic biology, they get upregulated and show up in your 2D Lamley gels because you need them to block omelet formation. So that's destructive complexity. So anyway, thank you for letting me say all this crazy stuff.
I had one comment on your example with the ATP synthase, because I found it very interesting, because you could use it. You explained it very well and said, I don't know if the increase in complexity in the specific case is the addition of the subunits. You said, I don't know if that's CNE, and you said maybe it isn't, maybe it has to be adaptive, or it could be adaptive and related to the morphological role of the supramolecular organization of the ATP synthase. But I find the example very interesting and compelling in the sense that if we think of CNE as being something that is neutral and that cannot be avoided, you can look at that diagram from the other side and see that all those branches where the ATPases stayed with the 17 subunits over 1 million years of evolution could be an argument to say that, okay, CNE might be existing, but it's not unavoidable, and it's not quantitatively overwhelming in that case, because you have, in that example, tons of a large evolutionary time in many different branches where there has not been any CNE. So I find the example very interesting because you can look at it you can turn it upside down and look at it from the other way around.
Yeah, I've had the same kind of thought. If these things have persisted for this long without that much change,
because if we go back to the analogy with the bureaucracy, from an intuitive point of view, what we have in mind is that bureaucracy and complexification of bureaucracy is unavoidable. That's a problem with communism, and that it will end up that way. That's a sort of a gut feeling. But if you apply that analogy, back to that system, then you have all these complexes, ATPases that have not suffered bureaucracy.
Yeah.
Bless them.
I think that's a great point. In the many explanations or many examples of CNE, there seems to be this runaway complexity in the trypanosome editing example, in the ciliate genome rearrangement example. There's no limit to where, I mean, apart from when selection finally acts on the runaway complexity, but there's seemingly no limit. And with these examples where we have seemingly pointless additions, I mean, if it ain't broke, don't fix it. If ATP synthase is working well, why do you add all of this crazy, really crazy complexity, and then suddenly just stop adding new things? It's good enough now, and it's not gonna. We changed it drastically all of a sudden, and now we're not going to change it again. And that's actually probably why I started changing my tune about ATP synthase complexity evolving neutrally. So yeah, that's a great point. Thank you.
Could I just add a small point on what's on the next slide? I think it's the very next slide. So this was your question about how long it would take to entrench more or less subunits. And I think it's kind of obvious that entrenching one subunit is in a sense easier than entrenching 10, but I would doubt that there's a significant difference between 1 and 2, and 2 and 3, and 3 and 4, etc. So if you can duplicate a gene, you can duplicate it and very quickly duplicate that. You can duplicate to produce whole families, so you can very quickly entrench large numbers. And if you produce multiple duplicates at the same time, then you could potentially entrench 4 at the same time.
So I agree. None of these are duplicates. So I should say that none of the genes added to any of these appear to be by gene duplication. Many of them are totally unrelated. We have no idea what they are, where they came from.
Yeah, I mean, do recall that gene duplication and subfunctionalization is only one mechanism of CNE, and something can get entrenched even just by being. So something really could have 5 or 6 or 10 or whatever things that get sort of entrenched all at once by CNE. It doesn't need to happen sequentially, and it doesn't get. And I think therefore it doesn't get sort of exponentially more difficult to do the more subunits.
Wait, it doesn't have to happen sequentially. I'm stuck on that because that's not how I understand fixation of things in population genetics. This is where I've always had. This is where I feel like I've learned a lot from Mike and Kerry that it, in a sense, fixation has to be sequential. Or you're fixing, or there's a what's it called when you're pulling along a bunch of deleterious mutations with an adaptive mutation, but you can't fix multiple things at once in a population. I don't think that's how it works.
Sure, you can fix multiple things at once in a population. Take 2 things, put them into one organism, kill everything except for that one organism.
Okay.
So if it's a small enough bottleneck, yeah, you can fix multiple things at the same time. And there's no reason in principle that the presuppressing entity can't be more than one subunit at the same time. 2 subunits can contribute to the same presuppressive effect that gets locked in by constructive neutral evolution.
I think we need to model it, Tyler.
Should model
I think maybe it's just a question of rarity or probability. Like it can happen. So it's just a question of what's likely, but yeah, you would need a model. And yeah, if you're talking about huge population crashes, then super weird things can happen. But if you're talking about things chugging along and fixing one at a time, that sequential fixation might be more likely, I would think.
I'm not sure why Ford didn't mention this so far, but I think there was a sort of older idea that in CNE, things needed to be fixed by drift. But it's not the case that they need to be fixed by drift. So it might be the case that, say there's difference in mitochondrial cristae or something, and one of the consequences of that is that there's some new proteins in there. Those proteins might be adaptive for some other reason, but then them becoming locked in parts of a new ATP synthase complex can itself happen by CNE. So it doesn't need to be the case that the origin of the subunit itself gets fixed by drift. It could have been fixed by selection a million years ago, and then later on, what CNE does is lock it into place in that interaction.
Yeah, no, B could
fixation.
subunit B could even have been fixed because it aided A in its function, but then it also suppressed possible, pre-suppressed possible mutations in A, which were deleterious otherwise. So, it could be mixed.
Yeah. I clearly that's true. So, okay. I'd have to, I can't think through that.
Yeah, it's hard to square that with the details of the A-to-B synthase case, but.
Well, I mean, like you can imagine a mutation. I mean, now we're getting into pleiotropy and multiple phenotype problems, which is hard enough to think about. So, I mean, you can imagine mutation that's beneficial for one reason, but neutral in terms of ATP synthase function. And you can imagine that the mutation is selected for because of its, I don't know, now this new, like you said, now this new protein is localized to the mitochondrial matrix, and it's doing a really good thing there, but it's also interacting with ATP synthase.
Yeah.
And you can see that being adaptive, and then you have a whole bunch of other things happening. Okay. But it can be modeled. And I'm really trying to find somebody that likes math and wants to model this. Because I think the problem is the people that like math either think this is too simple either too simple or too complex of a problem, or too esoteric, and everybody seems to be avoiding it.
Yeah, Mike gave up the last time I talked to him.
I mean, Mike likes the idea. He just doesn't. I mean, he wants someone else to do it.
That's how much you like it. Thank Jeremy, and see if we have some questions and comments that would be for the general discussion. I can see that everyone is exhausted, I think, in this room. I'm not sure about the Zoom, but yeah, I mean, maybe there are some more general questions or comments that people would want to make. Nobody dares.
Now, just a comment on modeling. I don't know if you know the work of Michael Lessig from Kerry. So he's a physicist, so he likes modeling. You might want to ask him, because he has a paper on adaptive ratchets and the evolution of molecular complexity. So that's not necessarily neutral evolution, but he's aware of the biology, and yeah, so that might be a person.
Yeah, you should share that person's name.
Yeah, I can send you the paper.
Yeah, great. Poor guy. Model for us. Model for us.
Exactly. You can do parallel emailing as our choice. I also saved the chat. There's some references and suggestions that we made in the chat. So yeah, we'll circulate the chat if you're interested. Any conclusion? Tyler, you've been doing all the talking anyway.
Well, I think we all sort of agree on what CNE is, which makes me happy. I think most of the disagreement still stems from questions like whether or not we need to invoke adaptation at the beginning of the narrative or not, whether it's okay to invoke adaptation in the middle of the narrative or not. And Ford and I sort of have taken sides to say that really we just sort of ignore the adaptive status of the presuppressor. But in other cases, it seems more important that the presuppressor is neutral. So that is something where people seem to still disagree. I'm trying to identify any other sources of disagreement within the discussion. I mean, Kerry and Jeremy and myself and Ford and Arlen all talk about the same types of things using different terms, which can be a little bit frustrating. So Kerry prefers buffer and pre-suppressor, and Jeremy prefers buffer and excess capacity. Kerry prefers, and Jeremy seems to prefer excess capacity. And Ford and I talk about pre-suppression, and Arlen talks about excess capacities. And sometimes I talk about capacities that aren't necessarily in excess. So, I mean, I think it's important to recognize that we're all kind of onto the same thing there at the end. I mean, I hope that becomes clear at the end.
Maybe pre-suppression is just a subfield of excessive capacity.
The other possibility that we're not considering, which is that the eukaryotic cell is really screwed up. You know, it's just terribly worse than the prokaryotic cell, and we might actually get rid of a huge amount of its complexity and still get a cell just as good, but it wouldn't perhaps have the evolvability that the eukaryotic cell has, and therefore we wouldn't be here to be talking about it. So in some ways, I mean, I'm a big believer in the anthropic principle, I guess, but it seems like, you know, our discussion does presuppose that we are capable of actually having this discussion, and it may well be that eukaryotes are the only way, and yet, you know, because of the evolvability characteristics that seem to be built into the eukaryotic cell and not, for instance, the prokaryotic cell. But it's all just chance because, you know, evolution doesn't look ahead. That's my piece.
Yeah, it could be deeply screwed up. I mean, I think that's the option people often ignore. They assume that there has to be some background, strong level of adaptation involved in almost everything they look at, and they're only willing to admit constraints when you give them good evidence that there must be some constraint. And they're only willing to admit neutrality when you assume, yeah, there was a population bottleneck. But actually, it could be worse than that.
Oh, it could be a lot worse. And I mean, I do think that molecular biologists and genomicists are all basically pan-adaptationists, and we sort of assume that, well, if it's there, it must be for something, and it must have been the product of natural selection. It's, in other words, an adaptation. And that's perhaps bullshit.
Maybe I can dare a comment, maybe a bit in that direction. I think that maybe we could oppose here modernism and postmodernism in the sense that looking both the pan-adaptionist view and also, you know, the neutralist or its opposition is obsessed with the notion. We said the cell is screwed up or it does better. So with the fundamental modern concept of progress. You know, like you would say in modern art, it has to be new, it has to be, you know, has to be an improvement. And in a sense, maybe we're too obsessed with that. And if we can combine the CNE and adaptionist view, you know, saying that something is screwed up, if it's there from a synchronicity point of view, if it exists as an organism, then it doesn't. Maybe we're too obsessed with the notion of progress and in a sense the evolutionary perspective. I'll go back to your example with the achlorophilitic plant. You can say that it's the. There are so many ways you can look at that as being, you know, a degenerated plant. You can see it as a symbiosis, but you can also see it as the plant being a parasite of the fungus, and in any, you know, whichever way you put on it, whichever name you put on it, we put that in a perspective, in a temporal perspective. But what matters is the synchronic character of the organism that exists at that given point, and the notion of something being more efficient or being screwed up or being degenerated is maybe obscuring in the sense that, again, I think we're thinking in a way that is too modern. We think too much with the notion of progress. And being a strong advocate of CNE does not, it was still in that perspective. Because we try to say, no, it's not like you think. We also obsessed with the notion of progress when we advocate CNE very strongly. And just to simplify and there I think I'm breaking through open doors. I think the question that is really interesting, or that is determinant, is the quantitative question. CNE is plausible. There are even very strong molecular examples that it's probably true. But the question is how much of it is going on. And since probably at any given time it's a mixed bag of adaptation and CNE, the question becomes really a quantitative question. How much of it and when? That was my comment.
We have never been moderns anyway.
I invite you to become the advocate of the postmodern biology that we all need to. I think there is a sort of a sad outcome of that comment, which is that maybe it has degenerated into a relative significance debate, but that's where it should be. I mean, we should be discussing not is or is this not a case of CNE, but how much CNE is involved in building the structure and how much
that would be progress.
Intellectual abuse.
Yeah, exactly.
You know, if the motto, the postmodern motto is anything goes, then it certainly applies both to CNE and to adaptionism. Anything goes. Whatever is.
Problem is there are a couple of other options that I may not be that happy with.
The other options?
Well, there may be other. You can also, if anything goes, then it's not just those two options that are on the table that are potentially like intelligent design and things like that.
Oh, okay.
So that's what like anything goes. I don't know.
No. I'm not saying.
Okay.
Sorry, I didn't say it in the sense that you can every narrative is equal. I'm not saying that. I'm saying from the biological point of view.
Okay. Yeah. Within that framework.
Yeah. If it's alive,
Yeah. Okay. Yeah. With that censorship.
Yeah.
Okay.
Do you know that slide that a lot of biologists like to give at the beginning of their talk, and they show the diversity of like life on Earth, all the different animals, and they say, I study what I study because I'm interested in this diversity, and then they usually start to talk about adaptation, like why each species is the special way it is. And so I guess I'm still just kind of reeling a little bit. I never thought this all the way through. So what we're saying is like complexity is just a runaway process. A lot of this diversity that we see at this very high level could be the result of a runaway process. That's wild.
Irreversible.
That's a great.
Yeah, it is wild.
Could be. Probably is.
Should we stop on this?
On this note, yeah. I think
we probably should.
Okay, so really thank you everyone. That was great.
Bye-bye. Thank you, Zoom people.
Thank you. Drink lots of wine for me.
Well.
Thanks.